Good morning, ladies and gentlemen. Welcome to Bern on this really beautiful day. My name is Martin Reist, and I serve the Swiss Confederation as Head of Animal Health and Welfare and member of the Executive Committee of the Federal Food Safety and Veterinary Office. I was a student of veterinary medicine 30 years ago. I quickly realized that veterinary medicine is really a complex matter. However, one aspect seemed relatively clear and simple to me: how to end the life of animals correctly. The protocols for euthanizing cats and dogs seemed very clear, so did the methods to correctly stunning and killing farm animals at the slaughterhouse, and there was not much mention about lab animals at all. Today, I know that I was completely wrong in my assessment. Not as far as the complexity of veterinary medicine is concerned, but as far as the correct and humane ending animals' lives is concerned. Dear participants on-site and online, dear members of the organizing committee, and dear expert group, it's a big pleasure to welcome you on this year's symposium on humanely ending the life of animals. For centuries, people have used animals for their needs for various reasons. We still do that today. We breed animals, we use them, and at the end, we kill them. Our society has been using animals for generations, and our generation is increasingly thinking about how we should improve our treatment of animals. In fact, we need to think about all the steps in the process. First, why and how many animals do we breed? Then, how do we treat them properly along the entire lifespan? And finally, how do we end the lives of animals? We know that we have learned a lot about how to breed animals and how to use them for our needs, but we have often failed to develop methods to end the lives of animals. This symposium is about sharing knowledge about methods to be improved and about looking for methods supposed to end the life of animals in the most humane way. The Universities Federation for Animal Welfare and the Federal Food Safety and Veterinary Office launched a joint contribution on this topic in 2018. In the first symposium in 2018, the focus was on CO2 in rodents. The second symposium in 2019 helped to elaborate priorities for the scientific investigation of possible alternatives to CO2. This prompted our office to award the University of Zurich a research contract to look for alternatives for CO2 for killing lab rodents. We are very excited to hear first results of this research today. In 2020, the third symposium started to extend the CO2 focus also on pigs, poultry, and other species. For this year's symposium, a group of experts defined 5 different topics which are supposed to be of high priority all over the world. It's animals in research, as before, but then also humane control of pests, slaughter of livestock, depopulation and emerging killing in case of epizooties, for example, and then finally the humane killing of aquatic species for food. As Federal Food Safety and Veterinary Office, we like to welcome you all who are attending the symposium online and on-site. And especially, we like to welcome the Universities Federation for Animal Welfare and the expert group and thank them for the careful organization of the symposium and their effort in animal welfare. I would also especially like to thank Otto Meissen and Josephine Korn and all the rest of the local team for their really great job in organizing this symposium. And a special welcome to Hugh Golich from the Universities Federation of Animal Welfare, whom I would like to give the floor to provide further information and open the symposium. Hugh is the Chief Executive Officer and Scientific Director of the University's Federation for Animal Welfare and the Humane Slaughter Association in the UK. He has been a major driving force in the establishment of this network and from the outset. So thank you very much, Hugh. The floor is yours.
Thank you very much, Martin. Thank you. So first of all, I'd like to say a very big thank you to the FSVO for their commitment to improving our knowledge and supporting research in this area. It's been a really fruitful and quite unusual partnership between an NGO and a government to make change in this area, and I'm really delighted to be part of this. I'm going to steal a moment of your time just to tell those of you who don't know very much about UFAW what we do and how we can support work in this area, I hope. So UFAW is an NGO. It's a charity whose vision is a world where the welfare of every animal affected by humans is maximised through scientific understanding of their needs and how we meet them. And it's that scientific part that I think is what makes us unique. We're all about evidence-based animal welfare. And we support that through scientific research and support for scientists who do animal welfare research. So we help discover what matters to animals and develop solutions to those problems. And crucially, try and disseminate that evidence so that it makes a real world difference. And that's what we're doing today, supporting this meeting with the FSVO to get the absolutely latest information about how to humanely end the lives of animals out in the world where it can make a real difference to animals. So we don't have as much money as the FSVO probably, but we do have some money to support scientific research. So if there are people here who need funding, they can certainly look to UFAW for some support to do research in this area. And we're very happy to support research in this area, as is our sister charity, the Humane Slaughter Association, when it comes to the welfare of livestock. We also publish a scientific journal, which you'll probably know is one of the places where an awful lot of research in this area is published. We're not afraid to publish research on some of these very difficult and controversial topics. It's now an open access journal, so I'd be very delighted to see some of the research that's being presented here today published in animal welfare. And finally, if you'd like to stay in touch with what we do, this is where you can sign up to our mailing list. So that's a little bit about UFAW. It just remains to say how delighted I am that there are so many people engaging with this meeting. There are 80-something people in this room probably, and more than 500 online, and it's amazing to see how many people care about what is a really difficult and controversial issue which would be easy to ignore. And so I'm absolutely delighted that people are engaging and looking to make real improvements for animal welfare. So thank you to everyone who's signed up to be involved in this meeting one way or another. So that's the introduction done. Now we move on to the important part, which is the science. And our first session is on the welfare of animals used in research, and it's going to be chaired by Dr. Patricia Turner, who's in charge of animal welfare at Charles River Laboratories. So I'm delighted to welcome Pat, who's been a long-term supporter of these meetings and work in this area in general. Welcome, Pat.
Well, thank you so much for that kind introduction, and thank you also to FSVO and UFAW for the opportunity to speak today. So perhaps also good afternoon and good evening to those online. My job for this session is really to set the stage for some of the exciting research talks that will come afterwards. So what I hope to do in this talk is to kind of review where we've come, some of the incremental changes that have occurred in the field, but also I hope to highlight some of the challenges that exist in what we know. So I'm going to mostly focus on some of the challenges around euthanizing rodents in research settings. some of our options for humane killing, effects of carbon dioxide gas inhalation as a review, some of the changes to the most recent AVMA guidelines, and then comments on CO2 versus other inhalant anesthetics for euthanasia. I'm also going to talk very, very briefly about humane killing of large research animals and maybe some opportunities for improvements in those areas as well. So I'll go ahead and get started. Getting this right, sometimes in North America, we tend to use the term euthanasia. In Europe, we tend to use the term humane killing. I mean, I'm talking about the same thing. I'll use both terms interchangeably through this. I think the important point is that through this, we're trying to seek a good death for animals, and that's a death that occurs with a minimum of pain and distress. And optimally, this is a death resulting from a rapid and irreversible loss of sensibility, followed by cardiac and respiratory arrest and brain death. There is a moral imperative for us to do this really well. And it's certainly a very high expectation of people who are working with these animals. And I'll say a little bit more about that later. And also, it's part of our social contract with the public for the ability to work with animals in science. So it is really important that we get it right. This is indicated, this importance for getting it right is indicated by the fact that this procedure of humane killing is referenced by all regional and national regulatory or compliance authorities and accrediting agencies. And I've just listed a number from different places. This is not an exhaustive list by any means, but just demonstrating that it's so important that every agency really defines this and emphasizes the importance of getting it right. So when we think about the needs for when the different sorts of conditions when we need to humanely kill research rodents, I just wanted to kind of set the stage for this because there's quite a number of variables that we need to think about. We need to think about the ages of the animals. So this goes all the way from newborn pups to pregnant dams to older animals. So we need methods that are available for a range of age groups. We need methods that are available for a range of conditions. Sometimes this is for animals that are maybe experiencing pain that can't be relieved, or sickness or injury. They may have reached the humane or experimental endpoint. It could be culling for population management. It might be one animal, could be a few animals, might be large numbers of animals. It can be emergencies like flooding that results in the need to humanely kill larger numbers of animals, or biosecurity breaks and large depopulations of 10,000 animals. So we need methods that are available for a range of animal numbers. Where these procedures occur, ideally in the vivarium procedure space, but in rooms that are in barriers, it might be in the holding room of for the animals if they can't be taken out, necropsy areas, on lab bench. So we need methods that are safe for a range of environmental conditions and exposures. And there are a range of individuals who may be conducting these procedures as well. So it's not just licensed veterinarians or veterinary technicians, could be researchers, students, and others. So we need methods that are simple to teach to a range of audiences. And then we have this issue of when does it occur? Sometimes, depending on the size of the facility, it could be multiple times a day. For smaller facilities, it might be only a couple of times a week. And so we have other issues associated with cost, convenience, and accessibility. And these are all really important as we think about what could be appropriate methods then that can be used in these settings. So common methods then that are permitted for humanely killing laboratory mice and rats, I've just summarized these very basically here. Barbiturate overdose is one of the techniques. This is a controlled substance, which means that it isn't widely accessible to everybody. It needs to be diluted typically for rodents, and there can be pain on injection because it's acidic. If it's given intraperitoneally, death isn't instantaneous with it. For that reason, we have inhalant anesthetic and carbon dioxide have been methods that have been widely used. Physical methods are also used quite a bit, so cervical dislocation or decapitation. The important thing to recognize, though, with some of these physical methods is that they have the most significant impact on compassion fatigue for the operator. So it's just people need to be trained really well in these methods as well. And research has demonstrated that even with very well-trained technicians, that sometimes the cervical dislocation doesn't occur in the place where it is expected to occur. So these are radiographs here just demonstrating, some fractures occurring in the thoracic area of animals that were meant to be cervically dislocated. And this study by Carboni et al. Demonstrated that, over 20%, around 21% of animals that were cervically dislocated had dislocations in inappropriate areas. So there are some challenges sometimes with physical methods too. So just this recap then, when we think about these preferences for humane death and the needs that we have for research rodents, when you start looking at some of these different areas here, carbon dioxide winds up checking the box for a lot of these different needs that we have for euthanasia of research rodents. And that's the basis for why carbon dioxide gas inhalation has evolved to be the most commonly used rodent anesthetic for euthanasia. That said, there are a number of challenges, of course, with using carbon dioxide for euthanasia. Carbon dioxide is a short-term anesthetic that was discovered well over 150 years ago, and euthanasia of rodents was first described with it in the 1920s. On the right side, we have a gas time curve there that shows rising levels of carbon dioxide over time. And we see insensibility in animals beginning to occur around 15 to 20% levels of CO2. The mechanism of action of carbon dioxide as an anesthetic is cerebrospinal fluid acidosis. That eventually results in stupor and coma of the animal or individual, if it's a human. The mechanism of action for euthanasia is apnea and cardiac arrest, and that occurs when animals are exposed to levels around 40 to 50% over a prolonged time. There are carbon dioxide sensors in the brain, so ion channels that are activated in people. These are activated around 5 to 10%, and that results in panic attacks in susceptible people. And we get the sense of breathlessness between 5 to 15% exposure level, so which can be quite distressing for people. And for animals, then we see gasping sometimes when they have this sense of dyspnea. And we also know that at higher concentrations, at over 45%, that breathing carbon dioxide can be painful. So it causes a stinging sensation because of carbonic anhydrase on the nasal mucosa and other mucosal surfaces as well. So what this means is that what the animal experiences when they are inhaling carbon dioxide really depends on the volume displacement rate, how long they're being held at different rates, and it's trying to find the spot that doesn't cause pain, but that doesn't leave them in this state of breathlessness and discomfort also for too long. And there are definitely potential for welfare concerns because of this distress and pain, and both of these are related to volume displacement rates. So what about other inhalant agents? There have been some early studies, and we will have another talk today on some of this, but early work, certainly with argon, which is shown here on the left-hand side, where the animals can go down this tube and get some Cheerios that are in the box at the bottom, but then the gas starts to flow, and you'll see this rat, when it goes down, it smells the argon, goes right back. So argon is not a good choice for rodents. It's aversive for them. The one on the other side is isoflurane, and early studies with this. So you can see the animal can sniff the isoflurane there, but he's busy eating his Cheerios, and he almost loses his righting reflex before he decides to turn around and go up. So he's almost insensible. You can see when he's quite wobbly when he turns around. And from these early studies, this provided an indication that perhaps isoflurane was not as noxious to laboratory rodents as carbon dioxide. So you can see he's quite wobbly there and decides to go up from it. That's at the first exposure. we'll talk about some additional work in a minute. I bring up the AVMA guidelines because they're used by a number of different regulatory authorities around the world, also used by ALAC in terms of a reference resource. And they revised and published guidelines in 2020, covered a number of things. They distinguished between various states of awareness in animals, not just rodents, but other animals. But in their most recent revision, they increased the carbon dioxide flow rate that was acceptable to use for laboratory rodents to 30% to 70% volume displacement rate per minute. And I don't have time in this short session to go into the research in detail, but this was based on a number of studies that were published after 2013. Most of these were single euthanasia trial experiments that happened in mice, where they saw either not a lot of significant differences in volume displacement rates between that 30 to 70% in terms of welfare impact, but also in rats as well. And again, similar to what I had described earlier, they noticed that there were increased panic-associated behaviors when rats were exposed to 20% levels of CO2 for long periods of time, while they had increased agitation, probably from pain, when animals were exposed to 100% CO2. So that 30%, again, seemed to represent sort of the best state for minimizing this potential for pain and distress when CO2 is used. So that's where those values came in the current AVMA guideline. Subsequent to that, a systematic review was published on the welfare impact of carbon dioxide and looked across at all the studies that have been published to date on that topic. And the overall finding of the systematic review was really that there was insufficient evidence to permit an unbiased assessment of the impact of a single exposure to carbon dioxide inhalation for euthanasia in laboratory rodents, because there were so many different indicators being measured by different groups, different flow rates and other things. Study design and other outcome assessment measures were quite variable, and missing information and other things that made it difficult to compare. Really, the conclusion was that more research really needs to be done in this area to be able to make some good recommendations about the use of carbon dioxide in laboratory rodents. So just kind of summarizing this, where sort of this knowledge is at, carbon dioxide is a general anesthetic. With slower fill level, animals likely become anesthetized before the concentrations reach those painful stinging levels that I talked about. However, there can be this moderate breathlessness that's uncomfortable during induction and maybe distressing. There have been a lot of these single trial euthanasia studies that have found no behavioral changes, but again, this gasping is seen quite a bit in studies before animals become insensible. We've also had some challenges with some of the studies that have been done with volatile anesthetics to date, anyway, with isoflurane and sevoflurane. Some concern that they might cause some stressful excitation on induction, and also that they can be irritating to mucous membranes. And some studies have shown aversion is stronger than for carbon dioxide when animals are re-exposed. And again, challenges because there's been a range of studies in this area. So it has meant it's been difficult to judge, again, because we have different study designs and comparators and things, and we're kind of looking at apples and oranges. It's been difficult to know whether volatile anesthetics are significantly better in all cases. And that's, again, been challenging. And as was alluded to at the very beginning, all of this has kind of come together with FSVO and UFAW and others with this paper in 2019 that really was emphasizing that for all species, when we're talking about alternatives to carbon dioxide, we need more research and more harmonization of aspects of study design for this. I also just, I want to also talk about opportunities for improving practices for large animal species too. So there's been a huge amount of interest in how we can best perform humane killing for laboratory rodents, but I want to also make sure that we don't forget all the other species we work with in research settings too. And one of the points that is made in the introductory comments of the ABMA 2020 guidelines for euthanasia is that the interests of the animal must be given equal consideration regardless of the setting. So when we think of research dogs, for example, the interests of those animals should be given the same consideration as for companion dogs. And you know what that means for the people working with them, but for the dogs themselves. And so something, I visit a lot of facilities in the course of my work, but also doing ALAC assessments and other things. And this is sort of an area that has been interesting to see and discuss at different places. But again, the ABMA guidelines recommend that for IV barbiturate euthanasia, that pre-sedation is recommended for that. Again, it's not because the technique of those performing euthanasia may not be done well, but it relaxes the animal. It means that if there's any discomfort from the injection, the animal doesn't feel it. They're not going to jerk back and maybe lose the venous access. Technical staff are more comfortable working with these animals. They don't have to worry about the experience, and we don't tend to see the excitement as much. But culturally, it isn't always the practice in veterinary medicine to provide a sedative, even in companion animal practice. In different countries in Europe, it's done quite differently. So this is something that we certainly try to emphasize when we're discussing this. So this is rabbits, dogs, pigs, primates, when we're working with them. The other consideration is for agricultural animal species. There are lots of pesticide or residue type studies that are done with agricultural species like cattle, pigs, and sheep. And those animals can't go to traditional waste streams because they have received radioactive tracers or other compounds. They are treated as biomedical waste, meaning they're often killed using captive bolt techniques, which I hear from technical personnel. It's quite upsetting for them that they've been working with these animals in research settings, providing a lot of care, and then suddenly a physical method that's quite immediate and shocking is used then for that humane killing at the end of the study. And so there really needs to be more consideration of the impact on personnel when determining the techniques to use in these settings again. So just a few comments again to get us sort of thinking about not just rodents, but that there are other considerations and we can be doing a better job in some of these other areas too. So just to wrap up, humane killing of research animals can be very difficult for those working closely with animals on study, regardless of the species. We didn't have time to talk about this much, but this means really people need to be really well trained. If they're doing caregiving, it's optimal if they are given the choice of whether they want to be present when those animals need to be euthanized, if there's enough staff and other things. So it really doesn't matter what species we're talking about. Euthanasia methods are fortunately still under constant study. We're going to hear about some of these things, some of these new proposals today. And guidelines are responsive to peer-reviewed research, so it does make a difference to keep doing research in this area. Study of the use of inhalant gas methods is ongoing for research rodents for the reasons I mentioned, and more research is needed in general, particularly for neonatal rodents, which may not be susceptible to methods that are approved for older rodents. And then opportunities also exist for improving humane killing methods for large animal species too. So thank you so much for your attention, and a few papers that will be available with my slides as well for those interested. And with that, I'd like to conclude and also then welcome our next speaker. So if we can, these can go down, and we can get the slides up for the next speaker. So thank you so much. And our next speaker for today is Dr. Charlotte Calvet, who will be talking about humanely ending the life of laboratory rats. And Charlotte is from the University of Zurich.
Thank you very much.
Thank you.
So good morning, everyone. Thank you very much for the opportunity to present my work on humanely ending the life of laboratory rats. So here are some pictures taken in the lab. So as Patricia mentioned before, there are discussions about the use of CO2 for euthanasia in rodents, because some studies show that CO2 might be aversive for rodents and that they might experience pain, discomfort, stress, or distress during euthanasia with CO2. And also one study in mice reported seizures with CO2 and also with other gases. So the remaining questions are to what extent rodents are exposed to pain, stress, discomfort, and distress, and this before loss of consciousness. But we still don't know exactly when loss of consciousness occurs. Is it close to the loss of motion or loss of posture? And what do animals experience before loss of consciousness? And we also don't know if other gases are less stressful than CO2 and if CO2 euthanasia can be improved. So this project was funded by the FSVO to humanly end the life of laboratory rodents. So today I will present the result on rats. So for this study, we used Sprague-Dawley rats, both males and females, and we used different gases. So we used 2 CO2 concentrations based on AVMA guidelines, so 30% and 70% CO2. We also anesthetize rats with isoflurane, and we also use carbon monoxide and nitrogen. So first, animals were handled and acclimatized to the animal facility for at least a week. And after that, we performed 2 surgeries. So one surgery for EEG implantation on the stereotactic device here, and one surgery for ECG and blood pressure implantation. And then 10 days after, so they recovered and they were placed in the plethysmography chamber you can see on the slide. So we recorded baseline data during 15 minutes, and then animals were euthanized or anesthetized. And we also recorded telemetry data and video recordings to see the behavior during euthanasia. After that, we collected organs, so we wanted to see the lung damage and also blood sampling to analyze stress parameters. So first, we saw an impact on the cardiovascular function. So here on this slide, you can see on the left side the 2 CO2 groups, then isoflurane, and then carbon monoxide and nitrogen. And you can see that with CO2, the heart rate is decreasing when the gas starts at time point 0 here. And it's the same with isoflurane, but with carbon monoxide and nitrogen, the heart rate first increase before they die. For the blood pressure, we saw an increase after exposure to all gases here. And this was also reported before by others, but we found differences with nitrogen because the exposure with nitrogen in this study was only for a short time. We also observed arrhythmias in all groups, not for all animals with isoflurane, but for all animals with the other groups. And it was faster with CO2 than other gases. Within a few seconds, animals had arrhythmias, and then because they died, they all had arrhythmias. And this has been reported to be painful in humans, some type of arrhythmias, so it might be stressful for the animals as well. Then we had a look at the respiratory function. So here you have the same groups, and you see when the gas starts at time point 0 that there is an increase in respiratory rate with all gases, also with isoflurane. But if you have a look now at the tidal volume, so how deep the animals are breathing, you can see that it's only increasing with CO2, carbon monoxide, and nitrogen. But with isoflurane here, it's first decreasing, and then during maintenance, it's a bit above baseline. And for the minute volume, you can see also it's increased after exposure to all gases. So what we can say is that with CO2, it can be due to an increase in the partial pressure of CO2. It's a compensatory mechanism to increase breathing. And it's fast and deep breath with carbon dioxide, carbon monoxide, and nitrogen. But the tidal volume with isoflurane is not increased. Then we had a look at the lung damage. So first, the airspace in the lungs. So this was in collaboration with a pathologist. And you can see here on the right side that the airspace is increased with carbon monoxide and nitrogen compared to CO2. So most likely there is a higher damage with these two gases compared to CO2. But for the number of erythrocytes, so the hemorrhage, there is no difference between the four groups. And then we had a look at the perivascular and intramural edema. And here you can see also that with nitrogen, it's a bit increased compared to carbon monoxide. Then we had a look at different responses to the gas influx. And here we compare to the baseline. So it's normalized per minute. The baseline is in gray. And you can see that with all groups, except carbon monoxide, sniffing and circling behaviors are increased compared to baseline levels. Then if you compare between groups, there are differences between the groups. And with 30% CO2 and nitrogen, animals sniffed more and circled more. But it's also correlated to the time to loss of motion because it's taking more time to lose motion, so they sniff and circle more. And then we had a look at gasping events, and we observed gasping mainly with CO2, with above concentrations, and most often before loss of motion. But we also observed a few gasping events with carbon monoxide and nitrogen, only in a few animals, and this was after loss of motion. Then we also had a look at the activity, and you can see here first you have the baseline for all groups here, and then the gas starts, and then there is a slight increase, but the increase was more pronounced with 30% CO2 in blue and nitrogen in green. And this increase in activity has been reported before. Then we assess the loss of physiologic functions. So first, this is not working. The loss of motion here on the left side, and it's faster with isoflurane and 70% CO2 compared to the other groups, as well as the loss of consciousness. I will explain later. And then the EEG became isoelectric a bit faster, it was a bit longer, with nitrogen and 30% CO2. It's the third graph. And then same for the cessation of breathing, taking longer with nitrogen and 30% CO2. And with 30% CO2 also, it took more time to die compared to carbon monoxide. But overall, for all gases, the animal lost motion first, or consciousness. Then the EEG got isoelectric. They stopped breathing more or less at the same time, and then they died. And this was also reported before. So now I want to show you some EEG analysis. So here you have the EEG raw signal for all groups. The beginning of the gas is the black line, and then you have the loss of motion with the red line. Here you also have the EMG raw signal, and this is a spectrogram of the EEG, so the different frequencies associated to the EEG. And if you take different ranges, you get the EEG waves, and here the percentage of each wave, so the dominance of each wave is represented, and the blue wave is delta, and you can see its dominance through the recordings for all gases. So here what I did is that I normalized to the baseline, so each wave is normalized to its own baseline, and you can see that when the gas starts at time point 0 here, there is an increase, and then there is a decrease around loss of motion. And this was for all animals. So we believe that this decrease in cortical activity could be associated to loss of consciousness, and also it's well correlated to loss of motion. So what I did next is that I compared the time to loss of motion based on video recordings to the time to lose consciousness based on this decrease in activity on the EEG data. And what you can see here is that the times are really close to each other, so there is no significant difference between the time to lose motion and this decrease in cortical activity. So it could be that loss of motion is a good indicator of loss of consciousness. Then, because seizures were reported in mice, we also had a look in seizures events, and we observed seizures only with carbon monoxide and nitrogen, but not with CO2, and this or isoflurane, and this for all animals. So all animals had seizures, and most often it was before loss of motion or consciousness. So they might experience this, and it can be painful for them. And also, the seizures were longer with nitrogen compared to carbon monoxide, and a bit higher for females compared to males. And this was also observed on video recordings, so it was well correlated. The EEG data was correlated to the video recordings. Then, finally, we had a look at stress parameter in the plasma, and we first checked the noradrenaline levels compared to baseline levels. Baseline levels were taken on awake rats on the vena saphena, and you can see here that there is an increase in noradrenaline levels in all groups, but it's a bit higher with carbon monoxide and nitrogen. Same for ACTH, you can see compared to baseline, there is an increase in all groups. And then we also had a look at corticosterone, but it was really variable between animals. And we believe that because there is a delayed release of corticosterone, that it's not a really good indicator for stress in this context, and that noradrenaline and ACTH are better indicators of the stress experienced by the animals. So in summary, so isoflurane is used as a standard anesthetics for laboratory animals, and we observe a fast loss of motion without gasping or seizures. And this was a second exposure because we did the surgery with isoflurane, so rats were exposed for the second time. Then 70% CO2 also resulted in rapid loss of motion and consciousness, similar to isoflurane, without seizures. But we observe gasping events in 89% of animals, and most often before loss of motion. For 30% CO2, it was longer to lose motion for the animals compared to 70% CO2, but we didn't observe seizures. And the problem with 30% CO2 was it was really variable. 50% of animals gasped, but yeah, it was only 50%, and it was also most often before loss of motion. With nitrogen, we observe an increase in heart rate and blood pressure that could be indicated of stress for the animals. We also observe seizures and a longer time to lose motion and consciousness. And the lung damage was more important than with CO2, as well as the noradrenaline levels. With carbon monoxide, it resulted in a fast death, but we also observe increased heart rate and blood pressure that could be related to stress and seizures. And the lung damage was also more important than with CO2, and noradrenaline levels were also increased. So if we can conclude something from this study is that carbon monoxide and nitrogens are not human alternatives to CO2 and should not be used to kill animals. And the perspective of this work, so we also performed this study in mice. Data is being analyzed, and we want to continue with combination of anesthetic gases with CO2 to reduce gasping events before they lose motion. And we will use two different strains, also males and females. Also in mice, it would be the same. And as I think we should also look at environmental factors, because this can be improved, and the way you handle animals is really important to reduce stress. And also, if they are euthanized in the home cage or in a new cage, it can be more or less stressful for them. Same for singular groups. We don't know what is best, but I think we should have a look at that. So I would like to thank everybody involved in this study, and thank you for your attention, and also thank you to the FSVO for funding this project.
So I think we have time for some questions, if anybody has questions. And I think at the back. Thank you for that very nice talk.
Thank you.
Very interesting.
Many thanks for the presentation. Very nice work. I just missed the concentration of the isoflurane you used.
So we induced at 5%, and then we maintain around 1.5%. Yeah, I didn't mention it, sorry.
Okay, thank you very much. And concerning, you were stating that seizure might be pain, but I don't think that based on the gas used and the pathophysiology, that seizure is pain. It's seizure.
We don't know exactly.
Exactly, but so it's important, because then I think you cannot take conclusions that are not supported with your data. And I would say seizure is seizure physiologically, and if they experience, and in any case, they cannot experience. We don't know if it's nociception is pain, but for sure you cannot demonstrate this.
It happens before loss of consciousness. They realize it. Yeah.
So it's fair to say that we don't know.
Yeah.
Okay, thank you.
Thank you.
Other questions? Okay, and if you don't mind introducing yourself and the institution you're from too, please. Thank you.
Thank you. Sorry, Julian Sparrow from Livestock Systems. Similar question to the other. You say nitrogen, but you didn't specify the concentration. We're sat in 80% now, so my assumption is you're saying 100% nitrogen, so zero oxygen.
Zero oxygen, yeah.
So did you perform an analysis on the residual oxygens that are in all the other cases?
We didn't.
No.
Yeah.
Because you'll get sort of 6%, 14%, and then zero oxygen.
Yeah,
no. Does that have would you feel that had an effect on the other gases or the behaviour of the animals?
The few studies using oxygen with CO2 are not in favor of adding oxygen, because it's taking more time to lose motion. So it's just, everything is longer. So I would say, probably it's not better, but we didn't do it, so.
In the 2020 AVMA guidelines, they actually removed that as a conditionally acceptable technique because it prolongs the time to loss of consciousness. So it's not recommended.
Hi, thanks for your talk. I'm Anna Lindholm, and I'm wondering why you had surgery in experimental design.
To implant telemetry transmitter to record all these physiological parameters. So we had to do 2 surgeries. So we wanted to correlate all parameters to see what happens when. That's why we recorded, as much as possible at the same time.
What would be your predictions for sevoflurane and the effects on the mice?
I would say, I hope it's, in mice, I don't know, but in rats maybe first, it should be close to isoflurane, so I would expect something similar, that there is no gasping, no seizures, and we can combine sevoflurane with CO2.
Hi, Jonas Knöll, Friedrich-Löffler-Institut in Germany. I'm wondering whether, with the EEG recording that you had, whether there can be artifacts from motion in there, or whether that it's easy to filter those out. So imagine if the animals have a seizure, they might be through the motion itself, not through brain activity, some artifacts.
Oh, it's also really visible on the video recordings that they have a seizure. So everything was
sure,
related.
but for then checking whether they are conscious or not, of course, it would be important to have the EEG without any artifacts.
Yeah. I didn't do the EEG analysis, but yeah, it was.
I think we'll hold questions at this point, but thank you again. That was a great talk. And so one more, please thank our speaker. And I'd like to invite Dr. Jasmine Clarkson from Newcastle University to speak about hypobaric hypoxia and hypercapnic hypoxia for euthanasia of research mice. Welcome.
Thank you very much, and good morning, everyone. So yeah, today I'm going to be talking about a project looking at hypobaric hypoxia as a potential alternative to CO2 in laboratory mice. So just to kind of put it in context, when we're talking about the numbers of rodents used every year, a very conservative global estimate is that 192.1 million rodents are used every single year for scientific purpose. And unfortunately, a prerequisite to their use is that they often have to be killed upon completion of the scientific work, or indeed at the end of their breeding life, for example. And therefore, I just wanted to highlight that it really does therefore represent the most widely used procedure. Now, in the UK, this is regulated by Schedule 1 of the Animal Scientific Procedures Act, and it permits 4 different methods, including overdose of an anaesthetic, exposure to carbon dioxide gas. The recommended fill rates are between 10 and 30%. So we had actually adopted a 20% flow rate in line with Schedule 1. Cervical dislocation, and also striking the cranium by blunt force trauma. Now, I'm not going to go into this in a lot of detail because Pat's done a fantastic job of highlighting this previously, but essentially we are all aware of the welfare concerns upon exposure to CO2 due to its ability to induce aversion, anxiety, breathlessness, and pain at higher concentrations, and therefore there really is an urgent need to find a humane alternative. So what we were looking at was the potential of gradual decompression, which has gained significant traction in recent years. Hypobaric hypoxia results from a process referred to as gradual decompression. So essentially, hypoxia results due to low pressure conditions, whereby as the atmospheric pressure falls, there's also a proportional decrease in the partial pressure of oxygen. And that's exactly why when people are climbing Mount Everest, for example, they'll often need supplementary oxygen. So therefore, it simulates a process equivalent to ascending to a high altitude. Now, importantly, and why people are particularly interested in this, is that hypobaric hypoxia is considered insidious in humans. People often don't know they're becoming dangerously hypoxic before they become cognitively impaired, and if you don't act accordingly, you will ultimately lose consciousness. And that's exactly why, for those of us that might have flown here, you were told to put your own oxygen mask on before helping others. So, obviously, we all appreciate that we can't ask animals how they feel, and therefore we've got to effectively determine how to assess their welfare, which can be trickier further in a killing context, and therefore we've got to, as animal welfare scientists, think about how can we determine their welfare during the killing process. So we've got to think about the time that it takes them to lose consciousness, their potential to experience negative sensations during that time, and then we can also use things such as drug interventions to look experimentally if, for example, pain relief or anti-anxiety medication cause an impact on any of these behaviours, and then that can give us a likely indication that it's either these behaviours are mediated through pain or indeed anxiety. So for this particular project, we took a very systematic and iterative approach with the overarching aim of evaluating the potential of hypobaric hypoxia as a high welfare method for killing laboratory rodents. So because we didn't know, first of all, whether this method was feasible to induce a non-recovery state and also do so in viable timeframes, we took the first experiment very much of proof of principle, and to protect welfare, we used terminally anaesthetized mice. Once we'd established that the method was effective at inducing a non-recovery state and in viable timeframes with minimal post-mortem damage, we then moved on to our behavioural trial, whereby we exposed conscious animals to gradual decompression and did a direct comparison to CO2. And we also used drug interventions such as pain relief and anti-anxiety medication to help us unpick whether these behaviours could be mediated by pain or anxiety. And then following that, we wanted to corroborate behaviour with neurophysiological assessments of consciousness in the mice, and that will help us better interpret the period of concern for animal welfare. So a little bit of details about the first experiment, the behavioural experiment with our drug interventions. We applied a slow overall rate of decompression. So we went back to the human literature and we decided upon a really slow 40 metre per second ascension equivalent, given that that's not associated with any ear discomfort or sinus pain in humans. We also then used, as I said already, our drug interventions. So we had analgesia and we had anxiolytics, and we also had mice receiving a saline control. And we, as well as comparing our decompression to CO2, we also had a sham treatment where the mice were just exposed to the killing apparatus, but nothing obviously happened. So what I wanted to just highlight with this figure is that because of the more gradual approach with gradual decompression, because we were taking a very slow ascension rate equivalent, gradual decompression does take longer for the animals to lose posture compared to CO2, which is illustrated by the red lines there. So that's the mean time for them to lose posture. The second thing that I wanted to just highlight here is the mechanism of action. So you can clearly see that as the atmospheric pressure falls, if we work out the atmospheric oxygen equivalent, you can see that oxygen is also reducing, which is obviously different to what happens with CO2. So with CO2, as we increase CO2 in the chamber, we also get a reduction in oxygen as well. So what we found from our behavioural experiment is that despite this longer induction time for the animals to lose posture, mice are actually performing normal species-typical behaviours. So for those of you that might not work with mice, they spend a lot of time exploring their surroundings, they'll rear, they'll dig, and they'll generally be relatively active. When they're more comfortable, they'll spend more time grooming. And therefore, the fact that despite this longer induction phase, the animals are performing these normal behaviours is really encouraging. And we also found no effects of our drug interventions on the expression of any of these normal behaviours. However, we took a very mechanistic approach to the way that we designed our ethogram. So we wanted to obviously just be purely descriptive rather than try and infer the likely consequences. So we did look at a lot of head-related behaviours, such as ear scratching and head flicks, for example. And with gradual decompression, although there were some mice performing these behaviours, there was no difference statistically in the likelihood of performing them, and there was also no effect of our pain relief or our anti-anxiety medication on their expression. And they were also occurring in the sham treatment where absolutely nothing's happening. So this was very different to what was happening with CO2. In line with Charlotte's talk, we got a lot of gasping with mice exposed to CO2. And what we also found is that was also mitigated through the provision of pain relief. Although the findings in terms of escape attempts isn't significant, given that obviously not all mice performed these escape attempts, which were a very vigorous vertical jump, what you can see is there is an indication there that mice are more likely to perform escape attempts when they're undergoing CO2 compared to the single mouse performing a single jump in decompression. And what I also wanted to just guide your eyes to with the CO2 is that you'll notice they're all saline-treated mice as well. So moving on to the final kind of experiment of the project, where we wanted to look at neurophysiological measures of consciousness. So we used EEG, and we wanted to obviously corroborate our behaviour with what's happening in the brain. So we obviously can't, unfortunately, put head caps on laboratory mice, and therefore we surgically implanted these wireless EEG transmitters. And we obviously allowed us to then really unpick the period of concern for animal welfare. However, like with all methods, it's not perfect, it does have limitations. We can't think of a transition straight from consciousness to unconsciousness. It's a much more gradual dimmer switch-like process. For this particular study, we conducted a survey in 2022 across the UK to find out how people are actually using CO2 in their facilities. And the previous findings that I've shown you were with top fill CO2, so we were obviously filling from the top given the density of the gas. But we actually found from our survey that most places in the UK are actually bottom filling, despite good practice being top fill. So we were interested in comparing those two CO2 methods with our decompression treatment. So we obtained 22 traces from mice undergoing one of the 3 respective treatment groups, and you can clearly see visually changes in the EEG signal with the state of consciousness. So during the baseline 30-second, when the mouse is indeed very conscious, nothing has happened, you can see a relatively high frequency activity. As the animal, upon the terminal treatment, and as the animal starts to be transitioned towards an impaired state of consciousness, we get this slow wave activity. And then upon, obviously, further on into the cycle, the signal becomes isoelectric. So that basically represents electrical noise from non-biological origin. So we wanted to do a formal time frequency analysis. And what this allowed us to do was essentially look at the power in each frequency band simultaneously across time. So on the y-axis, you've got frequency. On the x, you've got time. The greater the colour, so the red indicates more power. And we've got our red arrows, which is the mean time for them to lose posture behaviourally. And then we've got our 3 different treatment groups. So we've got bottom fill CO2, top fill CO2, and then decompression. And essentially what you can see is that the peak power in the spectrogram at baseline is around about 8 to 9 hertz, so very much in the alpha frequency range, which upon application of CO2, you see this gradual transition from alpha through theta and into delta. And if you look at where the delta peak is occurring, it's actually occurring after the animals have lost posture. This is very different to what's happening in decompression. Again, we've got this peak power occurring in the spectrogram around about 8 to 9 hertz in the alpha frequency range. And instead of this gradual transition through our frequency bands, we're seeing this really sharp step change in peak power, whereby peak power is very much in this slow wave activity domain, and therefore suggests impaired consciousness and would indeed suggest cognitive impairment. However, we've got to obviously look at what the animals are doing during this time. So essentially, we wanted to look at, we had a whole range of different behaviours, so they're not all on here for visual aesthetics. But essentially, what we found is that respiratory-related behaviours indicated by the right-hand circle there, such as first gasp, and ataxia, for example, and abnormal breathing, are all occurring during slow wave activity when the brain is showing that these animals have an impaired state of consciousness and are indeed likely cognitively impaired. With CO2, this is not the case. Again, we've got a whole range of behaviours, but essentially the animals are showing respiratory-related behaviours, ataxia, escape attempts and loss of posture, which are all occurring before the brain has transitioned into this slow wave state. We also wanted to obviously look at total counts of behaviours as well, given that they can tell us of an idea of cumulative harm during this time period. And very much in line with the findings that I summarised from the behavioural trial, again, we found more gasping when mice were exposed to CO2. We also found more escape attempts in CO2 compared to decompression, and there's a slight indication there when you look at the table that bottom fill somewhat results in more escape attempts. But once again, we found relatively normal species behaviours occurring during this longer induction phase during decompression. So again, they were rearing, they were grooming, which again is really encouraging. However, we did find in this particular study that mice were more likely to ear scratch when they were exposed to decompression compared to CO2. However, you can see there from the table that not all mice undergoing decompression ear scratch. And when we combine that with the fact that our sham mice were also ear scratching, and also the fact that our pain relief and anti-anxiety medication had no effect on its expression, means it's unlikely to reflect pain or anxiety. So just to summarise the neurophysiological key findings, I think it's really clear from the spectrograms that brain activity looks very different between gradual decompression and CO2. With decompression, we have this dominance of slow wave activity occurring really early on in the cycle. So it's occurring 105 seconds into the cycle and they don't lose posture till about 300 seconds. So it is occurring a lot sooner than loss of posture. And it's also occurring before they have respiratory-related behaviours, and therefore it could be associated with reduced negative sensations owing to respiratory distress. This is different, obviously, to what's happening in CO2. Instead, with CO2, we're seeing slow wave activity is actually not occurring till after the animals have lost posture, and they're obviously associated with respiratory behaviours, ataxia, and also escape attempts during that time. And therefore, there is an indication that these animals are more likely to experience negative sensations such as breathlessness due to respiratory distress. Now, I don't know how obviously relevant this is in a wider context, but at least in the UK, some facilities often turn the gas concentration up when the mouse has lost posture. So they'll induce at 20% of the chamber volume per minute until loss of posture, and then they'll turn the gas flow up. So this evidence is particularly concerning given that the brain activity is not yet fully in a slow waveform domain, and therefore there is this potential for them to experience nociceptive levels of CO2 before they're fully unconscious. So I hope you all kind of agree that gradual decompression could offer major refinement in the way that we kill millions of laboratory mice worldwide, and as such has direct implications for a whole range of different legislation and policies. However, working in this area, as a bit of a final note, over the past 5 years now, there really is this need for us to standardise the way that we kill laboratory rodents, not just in terms of CO2, but also cervical dislocation. And we really need to start thinking about developing quality controlled training programmes that everyone then sticks to the same guidance. So with that, it's very much not just me on my own. It was a hugely collegiate project. So I would like to thank my supervisory team, Dorothy McKeegan, Jess Martin, and Matt Leach, and also Julian Sparry, who was the genius behind the box development. So with that, thank you very much.
Thank you so much, Jasmine. I think we have time for 1 or 2 questions. Yes, so I think there's a mic. Again, please identify yourself and your institution for those online.
Kees Schepens from the Netherlands. I'm a veterinarian working with pigs. I would love to know whether you have any indication whether the ear scratching is relating to popping of ears.
It's a really difficult one to answer because my sham mice, when they were just placed in that environment, were also ear scratching. The data I've presented here was from one strain, Now, we've done a final experiment in a different strain, and I haven't analysed the data, so please don't hold me to this. I suspect it could be a slight strain-related effect, whereby that strain performs that behaviour. Because, yeah, as I say, our pain relief had no effect, our anti-anxiety medication had no effect, and also the sham mice were also doing it. So it's a bit of a tricky one to kind of work out exactly why they're doing it.
Great, thank you. Yeah, just any other, yeah, so one other question.
Alexandria Hicks Nelson, veterinarian from Fred Hutch Cancer Center. I was curious if you did any histopathology on any of the organs?
Yeah, so the first experiment that I obviously didn't end up having enough time to go into, the proof of principle, we did a full histological assessment and also post-mortem. So that is actually published. And we've also got some work currently been underdone as well with our slower rate of decompression as well, looking at exactly that between CO2 and also decompression, so that you will hopefully see that relatively soon.
Great, thank you so much.
Thank you.
Again, thank you for a great talk. And I'd like to call our next speaker up, Maya Bodner from the University of British Columbia in Canada. And Maya is going to be talking about refining the drop method for isoflurane induction of mice.
Perfect. Thank you so much for the introduction. Good morning, everyone. Today I'm going to be presenting on some of our recent work on refining the drop method for mouse isoflurane anesthesia. We've already had a bit of an introduction about this, but I want to go into it once more to remind you about a bit of a background of the killing of laboratory rodents, especially in the context of relevant guidelines. So in Canada, the Canadian Council on Animal Care states that humane killing methods must aim to minimize pain and distress, fear and anxiety, be reliable, reproducible, irreversible, simple, safe, and rapid. And many other national guidelines state something of a similar nature, with ultimately the method that you're using depending on a variety of factors. For example, the degree of suffering caused to the animal, the purpose of killing, for example, if tissues need to be collected after experimentation, and of course, the safety of personnel. That being said, there are a variety of accepted or conditionally accepted methods, such as those that are physical, the use of injectable agents, and inhalant agents. But generally speaking, the use of gaseous agents is preferred, as we can limit the restraint and handling of rodents, which usually aren't trained to undergo restraint, as well as these methods typically requiring less highly specialized training. That being said, the vast majority of laboratory rodents are killed with carbon dioxide, CO2, which we know there is evidence suggesting this to be aversive. So there are many euthanasia guidelines that do recommend anesthetizing mice with inhalant anesthetics, typically isoflurane, before administering carbon dioxide. And typically this is done with a vaporizer, which converts the liquid anesthetic into a gaseous form that can be inhaled by the animal. However, not all facilities have access to vaporizers, so this may be preventing them from refining their current mouse euthanasia or killing methods. However, there is a well-known alternative that's commonly used in fieldwork to anesthetize wildlife, when obviously there is limited access to vaporizers. And that's called the drop method, where we're essentially introducing a fixed volume of anesthetic into the induction chamber, typically by saturating a cotton pad or gauze with the liquid anesthetic and introducing this into the chamber. As I mentioned, this method is commonly used to anesthetize wildlife, but it has also been successful in anesthetizing lab mice, with some evidence showing that mice find this method to be aversive at standard concentrations of 5% isoflurane. However, we also know that lower concentrations, ranging from 1.7 to 3.7%, are suggested to reduce aversion, but this has only been assessed using a vaporizer so far. So in 2 different studies that we've done, I'm going to present to you today, we have aimed to refine this drop method by first figuring out whether we can actually successfully anesthetize mice within these lower ranges of isoflurane concentration, and secondly, we aim to assess aversion to this method. Which brings me to the objectives of our first study, which was to assess mouse behavior and insensibility in response to isoflurane exposure with the drop method using these lower concentrations. To carry this out, we used 27 surplus mice that we randomly allocated to one of the following concentrations. Our next step was then to determine how much volume of isoflurane do we need to achieve our desired concentration. And you may recall this formula if you've taken an introductory chemistry course. It's the ideal gas law. It's a simple law where we can combine all of these relevant variables to give us the volume needed to obtain this desired concentration. And you can use this depending on the specific parameters of your induction chamber. And ours looked something like this. It was a standard 5-liter container that we had adapted for the drop method. So you see a syringe up top that's dispensing the liquid isoflurane onto a cotton pad below. You'll see that we also fit a glove into the side of the induction chamber. You might be wondering why. This is so we can assess for anesthetic depth without having to open the lid of the chamber. This is really important, not just for the safety of personnel, but to ensure that no isoflurane is dissipating during the induction process. During induction, we also recorded stress-related behaviors as an effort to measure aversion, as well as several measures of insensibility. So what did we find in terms of our times taken to reach these measures of insensibility? From left to right, here are the times taken to reach ataxia, recumbency, loss of righting reflex, and loss of pedal withdrawal reflex. And we found that all mice were able to reach a surgical plane of anesthesia with concentrations as low as 1.7%. On average, as concentrations increased, we see this decrease in the average time it takes to reach these various measures, as expected. But moving on to our stress-related behaviors, we actually only measured one of these or observed one of these with a high enough frequency to allow for meaningful analysis. And that's rearing behavior, which is shown here, and its frequency and in 15-second intervals relative to the time that we administered isoflurane, with time 0 showing you our baseline values that we recorded before administering isoflurane. Unfortunately, we didn't detect a significant effect between isoflurane concentration and rearing behavior. But what's really important to note is that the performance of this rearing is consistent with onset of anesthesia. So as mice begin to become ataxic, this is very likely impacting their ability to express these stress-related behaviors, meaning perhaps this isn't the most reliable indication of aversion. So we aim to assess aversion using a different approach in our follow-up study. And this was through using a light-dark conditioned place aversion, or CPA paradigm. And these are common paradigms used to assess the aversive properties of drugs, where we're essentially conditioning an animal to associate a particular stimulus with being in a certain environment. So in our case, we're taking advantage of mice's innate preference for dark spaces and pairing this with isoflurane exposure. That way, when the isoflurane is removed, we can actually assess aversion just by looking at how much time these mice are spending within the dark chamber. So to carry this out, we first habituated our mice to our apparatus, as seen, and they then underwent what we call an initial preference assessment. There's no isoflurane exposure. They have 15 minutes to freely explore this apparatus. Mice were then conditioned for 2 minutes on the dark plus isoflurane side and on the light side, alternating each day for a total of 12 sessions, 6 sessions on each side. This is considered the amount necessary for conditioning to occur. However, we wanted to refine this procedure by giving mice the choice to enter the dark chamber. So mice were placed in the middle chamber, and you see that we have guillotine doors that allow us to control the location of the mice. The guillotine door to the dark chamber was lifted. If mice chose to enter this chamber, they were exposed for 2 minutes or until onset of recumbency. However, if they chose not to enter within 45 seconds for 2 consecutive trials, we considered them to have reached conditioning criteria. So this way, we're giving mice an opportunity to show us that they've been conditioned sooner than the standard 6 sessions, which may potentially reduce the number of forced exposures they have to isoflurane. Finally, mice underwent a final preference assessment. Again, there's no isoflurane exposure. And through this, we could calculate an aversion score, which is taking into account the time that mice are spending within this dark chamber in the final versus initial preference assessments. And since we were giving mice agency in entering this chamber, we were also interested in recording their latency to do so. So what are our aversion scores telling us? I'll preface this by saying that a more negative or decreasing aversion score indicates heightened aversion. And we certainly see that this is the case as isoflurane concentration increases, with a particularly big shift within the 2.7 to 3.7% concentration, where the score falls from -25 to -116, which is indicating to us that there is definitely learned aversion occurring at 3.7%. So if we take a step back and look at how we tried to assess aversion in our first study, we were just looking at acute behavioral indicators of anxiety during the induction process. The use of the aversion score in the CPA paradigm is giving us a completely different understanding, a different story in terms of assessing aversion, where we can rely on the animal's memory of their experience during these repeated isoflurane exposures. And finally, we took a look at their latency to enter the dark and isoflurane chamber across sessions. Here, mice are plotted individually with, on the y-axis, we have our latency to enter. On the x-axis, we have our conditioning days. We'd expected for this to increase across sessions, but we only noticed this to really be the case in the 3.7% treatment, where you see some mice do have this pronounced latency to enter in the later sessions, which is consistent with what we saw with our aversion scores. We also had one mouse in this treatment successfully reach conditioning criteria in fewer than 6 sessions. So now I just want to discuss the broader implications of this work. Our first study demonstrates that the drop method is certainly effective, that we can anesthetize mice with concentrations as low as 1.7% isoflurane. We've also demonstrated the practicality of implementing this method. When you use the drop method at 3.7%, you can actually achieve an induction time similar to that of using a vaporizer at standard concentrations of 5%. Finally, with our aversion scores that we recently looked at, we've shown that the drop method is minimally aversive at 1.7 and 2.7%, demonstrating that we can use these concentrations to refine our current killing practices for laboratory mice, and this may be particularly useful in facilities that are looking to do so, but perhaps don't have access to a vaporizer at the moment. That concludes my presentation. I want to thank you so much for listening. I also want to thank all the undergraduate research assistants that helped with data collection, all my collaborators, UBC animal care techs and veterinarians, and of course, all of the mice that are involved in our research. Thank you.
I think we have time for one question.
Perfect.
Right now. So if there's any pressing questions that anybody has. And if not, we'll
save it for the panel?
Yeah, save it for the panel session
Perfect.
at the end. Thank you
all.
so much, Maya. Okay, and our last speaker for the session is Kenzie Schwartz from Stanford University in the US, who'll be talking about Etomidate for euthanasia of zebrafish.
Great.
So welcome.
Thank you so much. Again, my name's Kenzie Schwartz. I'm a veterinarian completing my residency in comparative medicine at Stanford Medicine in the United States. And I'll be giving a short talk on evaluating Etomidate as a method of euthanasia in zebrafish, adult zebrafish. And more specifically, I'd compared 2 separate concentrations of Etomidate as well as 2 separate densities of fish within a single concentration. And so if you're not in research, a little background, what a zebrafish is. It's not a zebrafish hybrid. It's not this invasive species found in the southern waters of Florida. That's a lionfish, which I used to get them confused. But it is this little small minnow found in Southeast Asia with those 5 horizontal stripes that give it the name zebrafish. And the reason this becomes such an important area of discussion is due to the fact that zebrafish are one of the most commonly used animals in research, behind the rodents, of course. A study done in 2017, a survey found that they're used in over 3,250 institutions over across 100 countries. So that equates to potentially over 5 million used annually, and that was done in 2017. So now we know these numbers are still expected to grow with such advances in gene editing technologies like CRISPR-Cas9. And the reason they're such great animals for research is they do actually share 70% of the same genes as humans, And so they're used for a number of different disease models for genetic diseases, cancer, muscular dystrophy, tissue regeneration. And they require very little space, so they're very cheap as far as husbandry goes, and they do breed very rapidly, and so there's a very high embryo yield, which makes them great for research purposes. And so if you work with fish, you realize there's many challenges, unique challenges we face when we euthanize zebrafish. One of the most important ones and most difficult to assess is assessing their aversion or pain. As we know within the scientific community, there's that whole debate as to whether they feel pain or how they feel pain. I think we're going to have a talk about that tomorrow. However, in the veterinary community, we do just assume they feel pain and we treat them as such. So then there's the challenge of how do we assess this aversion? The most easy to use and most commonly used is just watching their behavior. So we watch for things like erratic movement, hyperlocomotion, piping at the surface, or twitching behaviors. And as we had just learned about conditioned avoidance tests, we can also use these for zebrafish. However, contrary to rodents, they actually prefer the light side, and then the anesthetic agent is added, and the time on each side of the tank is then evaluated. And you can also measure cortisol, which is a stress hormone, in that water. This, unfortunately, can't be used when evaluating Etomidate due to the fact that Etomidate will inhibit the synthesis of cortisol. Two other really important things to consider is the ease of use and human safety, just due to the fact that there's so many different personnel who are euthanizing the zebrafish and they aren't always properly trained. And which brings me to our leading methods in euthanasia in the United States, at least. Our most common method is using MS-222, tricaine methane sulfonate, which is a powder that requires the use of this fume hood, seen right here, which it's due to the fact that it causes skin, eye, respiratory irritation, and chronically it can cause retinal toxicity. And it also requires the addition of bicarbonate in order to increase the pH due to the fact that it is a very acidic powder. And there are numerous reports out there that suggest that it's very aversive to the fish. Another common method we use in the United States is rapid chilling, in which they're placed in ice water baths such as that one at 2 to 4 degrees Celsius. And again, there's multiple reports of this being aversive, and it is also extremely dependent on the life stage of the zebrafish, which I also want to bring up most of these methods are, depending on whether they're an embryo, juvenile, adult, egg, any of those. And then here's just a list of other common anesthetic agents that are currently being evaluated as other potential methods for euthanasia. Again, I just want to bring up the fact that all these different methods can be life stage dependent of the zebrafish. And so I just want to show you this paper that looked at the percent recovery according to age of the zebrafish and the time of exposure when using rapid cooling. And so they had essentially just found that the younger fish required much longer times of exposure than older fish. And fish that are at least 15 days post-fertilization are effectively euthanized when immersed for at least 5 minutes, and 30 seconds was not long enough. So what is Etomidate? The agent that I would like to talk about today, it's a non-barbiturate hypnotic agent. It's used for induction in mammal patients for anesthesia. It works by activating your GABA receptors, which are a neurotransmitter in the brain. It's generally very easy to use. You don't need a fume hood to prepare it. Generally safe for humans, clear color, so you can actually observe the fish in the tanks, and it works relatively quick. And so again, I had 2 separate aims in this study. The first aim was comparing 2 separate concentrations that are commonly used in the literature, so 6 mg per liter versus 10 mg per liter. And I also wanted to compare 2 separate densities of fish. So I euthanized them at 10 fish per liter as well as 5 fish per liter within a single concentration of 6 mg per liter. And they were immersed for a total of 30 minutes, and then a table tap was performed every 5 minutes to assess for a startle reflex. And then the fish were placed in a recovery tank for an additional 60 minutes to monitor for any signs of recovery. And then rapid chilling was performed as a secondary method to ensure that they were dead. These tanks were all video recorded, and then they were assessed by 2 blinded observers who were trained in zebrafish euthanasia and had lots of experience with it. And these are the specific parameters that were monitored. The first one was the loss of the righting reflex, so that ability to right the body orientation within the water. The second was undulation cessation, so loss of any movement forward, essentially immobility. The third was cessation of operculation, which is essentially respiratory arrest, loss of any of that buccal pumping. And then the last assessment was loss of that startle reflex, which we assessed by tapping the table every 5 minutes. And I just wanted to show you a video of what that startle reflex looked like. That was performed every 5 minutes, and boom, and then the fish would just kind of jolt forward. And so for my first aim, we had found that the higher concentration, 10 mg per liter, actually had shorter times to that undulation cessation, cessation of operculation, and loss of that startle reflex than the 6 mg per liter. You can see based on the graph that it wasn't an extreme variation, but it was significant. And I just want to draw your attention to the fact that the startle cessation did take upwards of like 10 to 12 minutes to lose. As far as density go, we did find that the 5 fish per liter had shorter times to loss of the righting reflex and to undulation cessation than the fish euthanized at 10 fish per liter. Again, not extreme variation, and that startle cessation lasted upwards of around 14 minutes. And I just wanted to show you this other paper who found similar things. They had evaluated all these different methods of euthanasia, which are all anesthetic agents, and they found a similar pattern in that Etomidate had a very short time to loss of that operculation, however, an extreme time until the loss of that startle reflex, upwards of what they had monitored, which was over 600 seconds. And so I also just wanted to show you a video of this very strange thing I saw. At about 20 minutes, most of the fish, not all of them, would start this kind of twitching behavior. And it didn't last upwards beyond 30 minutes. It lasted maybe 5 minutes. There was extreme variation there. And so I do want to just bring up the point that Etomidate does cause seizure-like behavior every now and then, some myoclonus activity in mammals that are anesthetized with it. And it's thought to be due to a buildup of glutamate in the system, in the neocortex. And so that's what we think is happening here. Again, we wouldn't know unless we had hooked up an EEG, and it just brings around the importance of using that secondary method to ensure that they are, in fact, brain dead. And so overall, we can say 6 mg per liter or 10 mg per liter was effective for euthanizing the adult zebrafish. A secondary method should still be performed. We also should still be evaluating all the other life stages of zebrafish. And density may, in fact, play a role. So just something to be aware of. Always euthanize your fish at your specific facilities' guidelines for housing densities. And further studies may also address alternative concentrations, immersion times, and densities. But we do believe it's a viable alternative for MS222 and rapid chilling. And thank you. That's all I have.
Great, thank you so much for a great talk. Do we have any questions, any immediate questions for Kenzie?
Hello, thank you so much for your presentation. It was really great to see. I'm from University of Gothenburg. My name is Lefteris. And I wanted to ask, have you considered to use EEGs to monitor the activity when you notice these seizures? So.
Yes, well, that's what I would like to do next. I think that would be a great next step. We didn't do that. This was going to be I hadn't planned to make this a project. I just had to euthanize a bunch of zebrafish at first. Figured I might as well videotape it.
Okay, thank you.
Great.
Any other questions? If not, again, thank you so much for a great talk. And I'd actually like to invite all the speakers from the session up to the table here. And we can open it up for some additional questions and take questions from online as well. So I guess first we can see if there's any more questions from the audience for any of our speakers. I'm going to ask a question then, if nobody has any while we're waiting. And then we can see also if we have any questions online. Oh,
So
we have a question here at the back.
Sorry, Alessandra Bergerano from the University of Bern. So I like the idea of the Etomidate, but we don't have your formulation. We have just the Lipuro, so the formulation with lipids in it, which will cause a problem to dissolve in water. So
do you have Metomidate available? Because that's another viable alternative that we actually aren't allowed to use in the United States currently.
No, so this can be an issue.
Yes, Metomidate might be a viable option in that instance.
Great, another question.
Hi, my name is Hans van der Vis. I work at Wageningen Livestock Research, and it's about using Etomidate for zebrafish. I was really happy that you mentioned that you want to use EG registrations to validate the method, because I think it is useful for you to know that we tested Etomidate on common carp, and when we administered stimuli to the fish, it didn't respond. But on the EG, the animal shows responses. So actually, in behavior, it gave the impression that it had lost consciousness, whereas on the EG, it was not unconscious. So I was really happy that you are considering using these methods as well.
Had you seen that startle reflex as well as the twitching behavior?
Yeah. To be honest, we did the work 18 years ago, so I can't remember all the details.
Great. Okay. Well, yeah, I think, the next study, we'll definitely try the EEG.
Thank you.
Thank you.
Great.
Hi. I'm Sonia Rey from the University of Stirling. Just some general questions. One is, how do you address the individual differences you find on fish and other animals and rodents as well? And another one is if you have considered to use some environmental enrichment when you've got them in chambers just to make them more comfortable, or just see how they change their behavior to improve the use of the anesthetics. And then just one thing for Kenzie is that fish don't have neocortex, so
correct.
It will be difficult to prove your hypothesis.
I agree, which is, thank you. If I could just address that. I agree, I had read that, and I'm curious. That's why I proposed I actually don't know what's causing that twitching or that kind of prolonged startle reflex, which is why I would love the EEG on there and see if we can try and figure what's going out in their brain due to that. As far as the environmental enrichment question, was that addressed for specifically fish, or was that? Okay.
When recommended, I know it is recommended to euthanize mice in the home cage, and so you could definitely account for that way, as long as, for using the drop method, for example, as long as you could modify the home cage and make it airtight to suit it for the drop method, that could definitely be a viable alternative, if you don't have access to a vaporizer.
Yeah, the same would be true for gradual decompression. Ideally, it'd be nice for you not to have to obviously remove the animal to put into another chamber. And the good thing about gradual decompression is you can obviously make the chamber however big you may need for a facility, and you can put the animals in their home cage to mitigate against additional stress that way.
In terms, I know you asked the first question about individual differences. There are so many different factors that could contribute to that. There's many studies done on individual differences in personality and how that can influence behavior. Anesthetic sensitivity can be influenced by factors such as sex, age, previous experiences, previous exposure to anesthesia, right? Whether animals are naive. So all of those definitely warrant further exploration and need to be taken into account.
Yeah,
Can I go? Or is there someone else? Okay. That brings me on to my question. I don't have any experience in rodent euthanasia, but I do with animals that are facing very stressful lives, that they're more difficult to kill humanely. And I'm wondering, is there a big movement in the rodent testing companies to improve the quality of lives to make your killing methods more humane? And then my second point I wanted to bring up, just because, Pat, you're from University of Guelph. I went to that university many years ago, and the central animal facility there, which uses a lot of dogs, what I loved about it is Dana Ben, who was the boss at the time, she organized with some students a dog walking program. So almost all of the lab dogs, we walked 3 times a week. And although it broke my heart to see these dogs in this research, I was really happy that at least we improved the quality of their life, that they were outdoors. Okay, thanks.
Charlotte,
maybe I can answer a little bit. I think handling is really important. If you handle them gently, it's better for them. They are less stressed. And if you pull mice or rats all together that don't know each other, I mean, it's stressful per se when you do that, and then you have to euthanize them. So I think this should be avoided. And I still see people doing that, and I think this should be forbidden everywhere. And we should really invest in handling, tunnel handling, rat tickling, and everything, because this might play a big role.
Yeah, I think just to jump in on that, I think there is a drive to do what we can in the kind of animals and science kind of world, and specifically rodents. I think we've seen a lot of talk recently about rat playpens, so rats have access to playpens on a certain number of times a week. But I don't know whether anyone's yet looked at whether that then affects how they kind of respond to killing. So yeah.
I'll mention I hadn't considered the impact of handling, but my thesis work is actually focused on refining routine handling procedures on rodents. This was kind of a carryover from my undergrad until I got to my master's. But I handle all of my mice with either a handling tunnel or with an inverted hut. And so perhaps that could be having an effect on their behavior expressed during euthanasia or their overall experience. That'd be interesting to look into.
I think we have one here. And then,
hello, my name is Nina Trimmel. I'm from the University of Fribourg in Switzerland, and my question would go to Charlotte and Ms. Clarkson due to the also additional fact that pain can be involved in euthanasia with CO2, but also maybe it can be painful with decompression method when the ears might pop. So I was wondering how viable you would think it is to also use grimace scaling for rats and mice, and also, of course, larger species, in context of CO2 euthanasia or decompression euthanasia.
Yeah, so I was very fortunate to work with Matt Leach. So he's done a lot of the rodent grimace scale work. The problem with the grimace scale in this context is it tends to be more of a longitudinal measure. You don't really see changes in the facial action units. So I was looking out for them, but it's too acute to kind of get that measure to look at. You can obviously look at very subtle components of it, but it's not something that has been found to be useful in this context because of kind of the more acute nature of it. As for the kind of potential for pain in the ears, the evidence we've got so far suggests that it's not a pain-related behaviour. Obviously, I wish I could say exactly why they're doing it, but one thing that I didn't have time to mention in my talk was that recompression, so obviously once you decompress the chamber, you have to recompress the chamber. And we know from humans, it's usually when you usually get ear pain and popping when you actually descend, which would be during recompression. So if we're kind of looking at it from the human literature as well, there is the suggestion there that it would happen during recompression and not decompression, when the animals would obviously not be conscious at that point.
Yeah, maybe I can add, we also had a look at the grimace scale, So I think you're right, it's like really acute, and it's hard to say also if it's pain or something else. And also with the setups we used in these chambers, you have different bias that make it hard to really access properly the Grimmer scale. For example, they were sleeping in the chamber, and then the eyes are closed and the score is really high. So it was really hard to assess it, and I think then it's not really a good indicator. So maybe stress parameters in the blood are better indicators.
I will just note that there is a 2023 paper out of the Motaroya's research group that demonstrates increased rat grimace scale scores with CO2 and actually a little bit higher with isoflurane on the induction part for euthanasia. Again, we know that rat grimace scale changes with anesthesia. And that's a challenge sometimes when scoring even pain after surgical procedures when we still have residual anesthetic effects. So I think that it's probably too early to say, and there's confounding factors that haven't fully been teased apart here. Yeah.
I was just going to bring you back to home cage versus euthanasia chamber and handling, and just really encourage some of you to actually do those experiments at some stage. And I just wanted to go back to a sort of counter argument that was put by, I think, Dan Weary, actually, who Maya works with, which was actually, if you woke up in your bedroom and it was filling with smoke, you might be even more stressed than in an unfamiliar environment. So maybe we're making an assumption there that's not necessarily right. So I just think it will be absolutely fascinating when your techniques are as refined as they can be to see what they are, and perhaps if they are still somewhat stressful, we might need to think differently even in that scenario.
There is another paper published out of Deb Hickman's group showing no difference when animals are euthanized in home cage versus a separate cage, but that's specific to the conditions of that particular study as well. So maybe suggestive of what you're saying, yeah. But that's a single study, yeah.
Yeah, I think that's kind of the next logical step for us. Obviously, we wanted to make sure that it was feasible and we had as much detail from an individual animal as possible. But that's very much kind of where we're hoping to go next. So yeah,
so
watch this space, hopefully.
You have another question?
Hi, I wanted to ask about the decompression method. What kind of equipment do you need and what kind of costs are involved?
I'm now looking at Jules. So essentially you need a sealed chamber and you have a vacuum pump. So you don't need any gases or anything as long as the chamber can withstand a very low pressure, and you've got a pump to essentially suck the air out. And obviously you need a lot of control over how quickly it does that. That's kind of what you would need. So it does have the benefit of kind of health and safety of not having gases. As for cost, I guess it just depends what you would need and the size. So yeah, that's probably more directed towards Julian at this point. Sorry, Jules.
Is there a microphone that we can have here? Thank you.
Unfortunately, it's very difficult to say because we built a laboratory version which is made out of acrylic, so you could actually see everything. I think a commercial version would be more straightforward in stainless and things like that. But until there's a market for it, I'll reserve on saying how much it might cost.
I think there was a question at the back, yeah.
Hi, thank you. So, question for the first two speakers as well. I'm wondering whether you could speculate what you'd expect if you get, if you're using hypoxia, let's say nitrogen, and mimic the time course of the residual oxygen that you had in the decompression. So I think that was the big difference between the two studies, that in the one there was hypoxia with nitrogen, but the residual oxygen wasn't, I think, directly measured.
No.
And in the second, in the decompression, we also get residual oxygen decrease, but with a controlled time course. So the question is, what might one expect if we have nitrogen, but mimic that decrease in residual oxygen, because it might be easier to do that technically, I think.
We have actually done a final, I feel like I'm always saying a final experiment on that particular project, where we exactly wanted to look at that. We didn't use nitrogen, we used argon, given the kind of evidence with nitrogen in laboratory rodents. So we used argon and we mapped as closely as we possibly could to that decompression curve. I'm still in the process of looking at that behaviourally. Subjectively, I don't know if it looks hugely different, but it's probably too early for me to say. So it was a very gradual induction of argon that mapped the oxygen equivalent of decompression, which perhaps hasn't been done quite that gradual before.
Thanks for your presentations, everyone. Raymond Anthony, I'm in the philosophy department at the University of Alaska Anchorage. Pat mentioned in her presentation that we should give due consideration to the personnel that may be called to use some of these techniques, and maybe if you could just reflect on what impact the work that you're doing has had on you.
Yeah, I think it's not a nice topic, is it? It is difficult some days, especially when you are exploring a novel method. You don't know what's going to happen, so you've got to prepare yourself for that. But it is challenging. But from my experience, I've been really lucky to be part of a really supportive group. We'd have very open discussions about that. So it was about seeking support from others when needed, and trying to kind of promote these open conversations about the topic, also within the group, but also externally as well with technicians in the department, for example. Yeah, but I guess that's kind of why there is a real interest in trying to find a high throughput alternative, because we know physical methods have greater compassion fatigue associated with them. So again, it's a tricky thing to kind of unpick. And yeah, I think there's definitely some further research that we can do on that. But from my own experiences, yeah, I've been very fortunate to be very supported.
Maybe I can add, I mean, I think I'm more driven by the fact that I want to improve the situation. So I'm doing the best to improve euthanasia. And now it's clear that we can use carbon monoxide and nitrogen because it was not clear for everyone. So then I know that people will do their best to finally find the more human method for euthanasia. So I'm more driven by that, but for sure when you see seizures, it's really not nice, and you want to avoid that animals experience pain or distress, and yeah.
Yeah, I'm also very motivated by just remembering that improving the lives of these animals is equally as important as improving their experience of death. And that's something that we certainly need to value. I've also been very fortunate, similar to Jasmine, to be surrounded by a very supportive network, very supportive community. My supervisors are extremely supportive as well. And addressing compassion fatigue is super important for us to be resilient in the work that we do. It's certainly very difficult, but I'm glad to be doing it, and I'm proud to be doing it.
I personally second everything they just said, and just more specifically to working with fish. Within research, they have probably way more regulations than the fish even used in aquatics and fish who are caught, and used for food. And so I just can always think back to that as the animals that I'm euthanizing have so many regulatory bodies, and it's being done in a proper way that's been evaluated with science.
Thank you so much. My name's Nikki. I'm from Nottingham Trent University. My question is more for Kenzie. So in your experimental design, you looked at 5 versus 10 fish. Are you thinking about potentially looking at the impacts if you culled them individually as well? Do you think there might be an impact in terms of stress responses if they're in a group versus by themselves?
Definitely. I think there's actually multiple papers out there that have looked at how housing density actually affects their stress, and they have found that fish that are at higher densities are actually more stressed. And so that's why I also only went up to 10 fish per liter. And so that's within our Stanford's guidelines, allows up to 10 fish per liter to be housed. And so it's just important to keep that in mind when you're euthanizing them. So,
yeah.
And I think the reason that we wanted to euthanize at higher densities is just due to the fact that when euthanizing in high numbers, time becomes a factor as well. And so
yeah, that was going to be my second question in terms of, is it actually feasible if animals are killed individually, is that actually a feasible method in real life?
Exactly. And with zebrafish, probably not. Probably not. Good question.
Great, thank you.
I'm going to take chair's privilege here to ask a couple of questions too, and then we'll go online as well. But Charlotte, in your study, you showed decreased tidal volume with isoflurane, which suggests perhaps breath holding compared to other groups. We know in human pediatric medicine, children given isoflurane find it unpleasant and they do breath holding. Did you want to comment maybe on that experience for the mice?
I know it's not used in human medicine anymore, so yeah. I don't know. Also, I would say it's a compromise of cost. But maybe sevoflurane would be better. We have to test it. So yeah, I'm looking forward for the next step of this study.
Yeah, great, thank you. And Jasmine, have you done this work in rats as well?
Not yet, but Sophia Bullock is here as well, and she's going to be exploring this in rats. So hopefully we'll have some nice data very soon.
And one of the concerns with prolonged period to death during the induction and process of euthanasia is the change in blood parameters. So this would be in a clinical setting when people are trying to collect blood afterwards. Have you done any validation of
We haven't,
blood
no.
parameters?
No.
Might be something to think
Yeah,
about,
absolutely. Something we can definitely have a chat about.
Great. Do we have any online questions?
One was for Jasmine, asking which strains of mice was used in these studies.
So the data I showed today was black 6 mice. We purely went with that strain because they're the most common in terms of transgenic models. They're often the one that people backcross onto. We have looked at bulb C on the proof of principle step, though. We used 2 strains to obviously look at post-mortem and feasibility of death in 2 strains. And then the final experiment where we've looked at argon mapped to decompression, we've gone back to bulb C and conscious mice as well. So although we obviously can't test every single strain, we're covering the majority that are used in research.
We have a question for Kenzie. Could the startle in zebrafish be spinally mediated reflex?
I guess potentially, because they're reacting to the vibrations in the water. Because I had also noticed if I had just tapped the tank itself, they wouldn't feel it. It wouldn't cause a reflex. So it was actually needing the table and the vibrations through the water. But again, so would they be brain dead after that, or is it just a postmortem reflex? Good question.
Any other online questions? Jess.
Jess Martin from Newcastle University. So this is more for Charlotte and Jazz. Do you think there is enough evidence out there now to support CO2 being removed as a method of killing?
That is a mean question.
Is there sufficient evidence?
Oh, Jess.
So Jess Martin, Newcastle University. Do you think there's sufficient evidence now to support the removal of CO2 as a method of killing for laboratory rodents?
You want to start, or I can start maybe? So really what I observe is gasping events, but not with all animals. And I would say, as we mentioned before, the environmental factors are really important, and maybe it can be improved like that. So it's not only about the gas, but everything around to reduce stress and to improve euthanasia in animals. So I would say there is not enough evidence at the moment to say we can further refine and improve it, and maybe we can conclude, but at the moment I would say no.
I would take the opposite approach, I
Okay.
think. I think I would say there is enough evidence. However, the worry and concern with banning it, I guess, would be what are you pushing people towards as an alternative? And I think there's still a lot of uncertainty as to what that would look like and what that would be. So we did a survey looking at what common methods people were using across the UK, and most people were cervically dislocating. But as Pat showed, there's a lot of variability. People aren't necessarily dislocating in the correct location. So that's something that I'm really interested in, and UFAW have kindly funded a small project for me to look at that a bit further, because I guess the concern is if you ban something, what are you then pushing them towards? And is that actually better if, for example, someone isn't confident in a physical method, but they're told, well, that's your only option for this particular scientific outcome? Are they going to be doing it confidently and correctly? So I think there is enough evidence, but I think it's about having an alternative at the same time, for me anyway.
If I can add something, maybe. I think also people are biased, because if you show them video of anesthesia compared to euthanasia, they will believe euthanasia is worse. But if you don't mention which method is what, so isoflurane or CO2, they will say both are the same and they feel the same. So I think we should really consider that the impact on us and not judge just based on that. And yeah. Well, I don't agree, but yeah.
Yep.
So I guess it's a follow-on for that one, really, probably mainly for Jasmine, which is your method would need to be authorised to become a standard method, at least in EU. It would need to be added to Annex 4 of the directive and Schedule 1 in the UK, and I guess probably in Switzerland too. So A, how close do you think it might be to trying to get that put into practice, and B, do you think the hurdle is too high to have a method accepted as a better alternative?
So we've started to have these conversations. The hurdle may not be as high as what we initially thought. So yeah, that's something that we are discussing at the moment as a project team, and we will be kind of addressing how we go about that, at least in the UK as first instance, and perhaps it's a case of looking at it locally, so even just kind of if we can get approval to kind of have it at Newcastle, and then we can kind of be a bit of a guinea pig and a pilot for how it works in practice, then perhaps it might not be such a big hurdle if we are kind of testing it out initially.
Good to hear.
Have you looked at the technique for neonatal rodents?
Not yet.
I mean, so there are
Yes.
lots of other little studies that are...
Yeah, it would very much be restricted to adults only until we had any evidence to suggest it would be a viable alternative for neonates, yeah.
Great. Any other final questions as we look to wrap up the session? And If not, can I get a round of applause, please, for our speakers?
Okay, everyone, thanks for coming back in. We're going to start the second session this morning with a bit of a change of subject. So this session is about, I'm going to use use inverted commas a lot, the control of pests this morning. So I'm going to do a very quick introduction to this topic, which is something that UFAW has been quite involved with, and then we're going to move on to two really interesting talks in this session. So pest control is a really different issue in terms of our attitudes to perhaps how we think of some of the other areas of killing animals which we're speaking about at this meeting. And there's an enormous range of attitudes, but I pinched this poster from I think the 1950s from the Canadian province of Alberta, which shows you one end of the spectrum of the attitude to controlling pests, which is almost to see it as an obligation. And Alberta has managed to keep itself almost entirely free of rats by really putting an enormous effort into controlling them almost at any expense. So this is one approach, and pest control is probably the largest deliberate anthropogenic human-related effect on wild animals. Obviously, we have other effects which are not intentional, but we kill millions and millions of wild animals because they're so-called pests or invasive species, or they're in conflict with us, which essentially really means they're an animal in the wrong place at the wrong time for at least some of us. So there are a lot of similarities with the animal welfare issues around control of pests, but also some really quite significant differences compared to how we deal with animals in research or the animals that we consume for food and so on. So I'll just try and highlight a couple of issues before we move on to the main talks. So obviously, the first thing to think about is why we control them. And I think often, quite a lot of people first think of their personal experience where they've had to deal with animals which have infested their house, for instance, and are perhaps damaging property. But we need to acknowledge that there are really some much more significant global issues with pest animals, which are a major threat to human health and food security and ecosystems. And also to remember that a lot of these problems are much more significant in the global south. So in many situations, I think many of us would accept that control of pests is unavoidable. We're going to have to do it to preserve global food security and human health. So the question then becomes whether we can do this humanely or not. And sadly, this isn't one of the issues where there's a very clear one welfare solution for a lot of these problems. What's good for human welfare in terms of controlling these animals is often tremendously bad for some of these animals' welfare. Many effective pest control methods are incredibly inhumane, and we really wouldn't consider accepting the kind of welfare impacts that some of these methods, like glue traps or anticoagulant rodenticides, which poison rodents over several days with attendant pain and suffering. We simply wouldn't accept those if they were proposed as something that was going to be done in a research scenario, for instance, where we look at a harm-benefit analysis. So there really are some really substantial animal welfare issues here. Another thing that's interesting is that pest control affects non-target animals. It's far from uncommon, sadly, to accidentally poison domestic pets and other animals with rodenticides, or to catch non-target species in traps, and often actually the welfare impact on those animals can be worse than the impact on the target animal because the method wasn't designed for these animals who accidentally come into contact with it. So that's another interesting problem. Also, killing is not always the only solution in these problems, and we're going to hear about some alternatives in this session. So there are other ways to control these pests, for instance, contraception, fertility control, translocation, moving them somewhere else, simply excluding them from the areas where they cause a problem, mitigating the impact, or even some people would advocate that we should just learn to live with some of these animals rather than trying to remove them from our environment. On the other hand, killing might not always be the worst solution. And I became acutely aware of this when the UK banned glue traps for rodents, which is a good thing, which I thoroughly support. But that then raised the question of what should we be doing to control rodents? And lots of people like to suggest that what you should do with a rat or a mouse who gets into your house is trap it in a humane trap and release it somewhere. And unfortunately, I don't think that's been very well thought through. And you can see even PETA, who are really strong advocates of not killing rodents, acknowledge that if you catch a rat and release it somewhere out in the countryside, it's likely to not do very well. It's either going to starve, come into conflict with the local indigenous rat population, or be predated. And they suggest maybe you should release it nearby, but I suspect that will not work well either, because it will either get its way back into the house that it was in the first place, or even if you block that access, they may well end up in other properties and someone else will kill them, quite likely in an inhumane way. So it's actually quite reasonable to argue that lethal control would be the better option in this scenario if we can find a humane lethal control method. I think what's really evident in this field is that we don't have enough evidence about a lot of the methods we use. What we'd ideally like to do when we talk about what's the best way to deal with any of these pests is to have lots of really good primary scientific evidence, do a nice systematic review and meta-analysis, and tell you unequivocally that this is the least inhumane way of dealing with these animals. But sadly, we don't have enough primary evidence in most of these fields. So an approach I wanted to highlight, which we'll also hear about in the next talk, which UFAW and others have supported, is a so-called 5 domains approach, which essentially takes what evidence there is and asks experts to look at that evidence and use their own personal expertise to assess the welfare impact using the 5 domains model, which lots of you are probably familiar with, of different methods to give at least some way of ranking different methods in terms of their impact. And for instance, this is an example looking at some of the rat control methods, which looks at, you've got 2 axes here because you look at the impact of the killing method and also the impact of everything else that you have to do prior to that, which might well involve trapping the animal, for instance, and look at the intensity of both of those. And overall, that will give you something approaching a score where you can compare methods. And if you look at this one, what it shows, this is an expert consensus from welfare experts, veterinarians, pest controllers, so a wide range of expertise. And what it concludes is that glue traps, unsurprisingly, are the worst impact on rats. And probably the lowest impact is snap traps. But you can see, which is number 1 on that graph, but you can see it's a very wide spectrum of impacts because a snap trap can work incredibly well and kill a rat instantly the moment it goes into the trap or near the trap, which is a very low welfare impact because death is instant. On the other hand, animals can become trapped and not die instantly and take a long time. So that's why you see this wide spectrum. But I just wanted to highlight that as an approach, which we may well hear more about in upcoming talks, and can be applied to novel methods to see whether they are genuinely looking like an improvement compared to how we assess existing methods. Common theme, which we heard in the last talk, we need to do more research about this. We need to know a lot more about the current methods, because ideally we would know enough about both the welfare impacts and, importantly, whether they actually are effective or not, because there's quite a lot of doubt about how effective some of these methods are. And then we can do what we do in the research scenario, which is assess the harms and the benefits to work out what we should actually be doing. And similarly, we need research to develop new methods so that we can assess them with this kind of framework. That's where I'm going to stop and introduce our first speaker. But it wouldn't be a conference if we didn't have someone who'd managed to catch COVID and couldn't attend in person. So sadly, Professor Naya Beausoleil is stranded in New Zealand with COVID, so we're going to be hearing her talk as a recording, which also, I'm sorry, means she won't be able to answer questions because it's also midnight in New Zealand at the moment. So hopefully we can play you her recording now. Fingers crossed.
Hi, everybody, and thanks so much for having me talk here today. I'm very sad not to be there in person, but after 4 years, COVID finally got me, and it's just a case of really bad timing. What I'd like to talk about today is how can new poisons kill more humanely? So just start off with a bit of a plan of what I'm going to cover in the next 20 minutes. First, I'd like to give a bit of general information about the control of introduced animals. These are vertebrate animals. So why these animals are controlled and how we use lethal methods to control them. I then want to spend a bit of time talking about pest control in Aotearoa, New Zealand specifically, and a large program that's been started by the New Zealand government to eradicate mammalian predators in New Zealand. Then I want to go on and tell you a little bit about some early thinking that we've been doing about how we can build animal welfare science into pathways for developing new or refined toxins for lethal pest control, and talk about how we can integrate the 3 R's into those pathways. And finally, I'll do a bit of a summing up. So in general, the control of vertebrate animals goes by lots of different names. We might talk about pest control, control of invasive animals, or introduced animals. This terminology actually does matter because it influences the way that we interact with those animals and the permission we give ourselves to treat them in various ways. But for our purposes today, really, all we need to think about is that we have a desire or a need to control animals that are in a place that we don't want them to be, or they're in that place in numbers that don't work for us. And there are various reasons that we want to control these kinds of animals. So those include for reasons of food safety or security. For example, you might think about rodents in grain stores or in food preparation areas. There are concerns about human health. So for example, zoonotic diseases. We might have concerns about the protection of property. An example might be in Australia where there are too many kangaroos in an area where we're trying to graze domestic animals. And in New Zealand, a really important one is conservation. So conserving populations of other kinds of animals, plants, or ecosystems. And conservation is a really important reason in areas like New Zealand, which have very unique flora and fauna and are very isolated from other incursions of these kinds of animals. There might also be concerns about the welfare of the pest animal itself. So, for example, if natural control of the pest animal populations has been reduced and so they're actually outstripping their own resources. So globally, this pest control relies heavily on lethal methods. We use a lot of different kinds of kill traps, and we use a lot of toxins. In terms of the kinds of toxins that are used, the anticoagulants are very commonly used. These are products that inhibit blood clotting, and also 1080, which is the other name for monosodium fluoroacetate. However, the use of toxins is becoming progressively less socially acceptable, and there are a number of reasons for that. So, for example, in New Zealand, there are perceived environmental impacts, concerns about things like water quality if we're spreading these toxins throughout the environment. There are cultural reasons for concern. The First Nations people of New Zealand often have concerns about managing pests using toxins. And then there are also concerns about the impacts of these toxins on non-target animals. So, for example, valued hunting animals like deer in New Zealand, or companion animals that get accidentally poisoned. So I just want to spend a little bit of time talking about the pest control situation in Aotearoa, New Zealand specifically. And I need to do this because on occasion I've spoken about this issue overseas, and people become really concerned when I talk about this idea of lethally controlling all of the mammalian predators in New Zealand. So it's really important to understand that in New Zealand we have no indigenous mammals at all, except for 2 species of bat. And we don't have any native mammalian predators at all. So all the control we do is on these pest or introduced or invasive mammal species. New Zealand has an incredibly unique fauna. There are many species of birds, reptiles, and invertebrates found nowhere else in the world, so they're highly valued. Since human colonization, numerous mammalian species have been introduced to New Zealand, and this includes 3 species of rat and also the house mouse. We have 3 species of mustelids, so stoats, ferrets, and weasels. We have the brush-tailed possum, which is shown in this bottom picture here. These animals were introduced from Australia to generate a fur trade, which turned out not to be such a great idea. They've been hugely successful in New Zealand. And then there are a whole range of other mammalian species that have been introduced. So things like rabbits, the mustelids were introduced to try to control the rabbit populations. We also have wallabies from Australia, hedgehogs, and a whole range of different birds, fish, reptiles, and amphibians. And these were brought either purposefully by settlers or came along with them accidentally. Collectively, these species have caused enormous environmental and economic impacts, and these have come about because they've preyed directly on our native animals or outcompeted them, and a large number of native birds, reptiles, and invertebrates have gone extinct since humans came to New Zealand. They've also destroyed habitat, or they now compete for grazing, for example, rabbits and possums competing for grazing with domestic livestock. And then there are concerns about animal disease. So, for example, brush-tailed possums can transmit tuberculosis to cows. And for anybody who knows anything about New Zealand, dairy cows are extremely important to our economy. So for conservation and animal health reasons, New Zealand finds itself being a world leader in lethal pest control. A couple of years ago, the New Zealand government set an audacious and ambitious objective to become predator-free by the year 2050. So we've got about 26 years to achieve this goal, which is to eradicate 7 mammalian predators. And those are the 3 species of rats, the 3 species of mustelids, the brush-tailed possum. Those are considered for this campaign to be predators. So you'll notice that cats are not included in that list, along with a couple of other things, and nor are mice. An enormous amount of effort and money has gone into ensuring that New Zealanders get behind this campaign so that we have continuing social license to kill millions and millions of sentient mammalian predators. And a lot of money has also gone into advancing technology to achieve this goal, this eradication. What's clear is that without lethal pest control, we won't save the remaining indigenous species. And it's also clear that while we rely heavily on toxins, the tools that we have currently are not effective to achieve this eradication goal. So we're not just trying to reduce these predators to small numbers. The goal is to get rid of every last one. And we know that with the tools we have right now, we can't achieve that. Some of the reasons for that are because we have large remote areas to control, that animals become shy of traps and also of some baits, or there's actually some developing resistance to baits in some places. There are concerns about using these toxins, which are general toxins, in areas where humans or companion animals might encounter them. And then there are also concerns about non-target effects on native animals. So we get accidental mortality and welfare impacts on the animals that we're actually trying to conserve. The government has invested heavily in far horizon technologies. So these are genetic control tools, for example, but the feeling is really that those are not close enough to being realized to achieve this goal. And so in the interim, the government has also put more funding into developing stopgap technologies, and that's really where the work that I'm involved in comes in. So one of those stopgaps is the development of novel toxins that are selective or that are more selective for a particular species or taxon. So this has led to a large research program that involves a multitude of different institutions and researchers from a whole range of different disciplines. We've got chemists, we've got social scientists, and I'm the animal welfare scientist on this program. And this precision pest eradication program has 3 overarching aims. The first is to discover novel pest selective toxins. So molecules or compounds that are toxic only for the species or the taxon of interest. So for example, that might be rodents or it might be rats specifically. to refine existing toxins so that they're more toxic for those target animals, so in some way synergizing some of the molecules that we might already be using, for example, or to refine existing toxins to be less toxic for non-targets, so for example, our native birds. So obviously my interest and involvement is the animal welfare aspects of this program. And we've developed a sub-team that's considering how we can build welfare science into these development pathways for novel or for refined toxins for lethal pest control. The overarching aim, obviously, is to reduce non-target mortality and welfare impacts, so for example, on our native birds. But we want to develop these new or refined toxins that are effective. However, we also want them to be less harmful for the target animals. Now, that seems kind of weird. We want to actually kill these animals. These are lethal toxins. So what in the world do I mean by less harmful? Well, what we mean is we want to reduce the intensity or duration of the negative impacts that the animal experiences before the point of irreversible unconsciousness and death. So the animals are still intended to be killed, but we want them to suffer less before they die. And these aims kind of beg a number of high-level questions. So, for example, less harmful than what? We've also got the question of how can we build scientific assessment of welfare impacts into these development pathways for new or refined toxins? And challenging, how can we know how harmful a new or modified toxin might be before it's fully developed? So we're quite early in this program of research, and we're early in our thinking about these questions. So I just want to make a few comments about what we've been thinking about so far, and to invite people to get in touch with me if you have some other clever ideas or thoughts about these questions. So we have these three kind of key questions: less harmful than what? How can we build scientific welfare assessment into our development pathways? And how can we know how harmful something's likely to be before we've actually developed and tested it? In response to all three of those questions, I would make a couple of general points. The first is that when we develop teams that are aiming to discover or to refine toxins for lethal pest control, we need to make sure we have the right people on the team. And so I would argue that it's really important to include people with appropriate animal welfare science expertise right from the start. And if you think about it, probably everybody's here has had an example where you've brought a statistician into your research team a little bit too late in the process, and the statistician is fairly annoyed because you've gone off and designed your study, you've collected your data, and then you've come to your statistician and said, here's my data set, now can you please fix it and make it answer the question that I want it to answer? It's really important that we have people with appropriate expertise on these teams right from the very start, right from inception and the design phases. The second point I would make is that it's important to use structured, science-informed approaches to assessment, because if we do this, we get comparable outcomes, and then we can then compare different molecules, for example, when we're deciding whether or not to progress them through the development pathway. Thinking more specifically about the three questions, the first one is less harmful than what? And that's a really interesting and challenging ethical question. How can we decide what our reference point is for continuing along a development pathway for a particular molecule or compound? And I'm keen to hear people's thoughts about this. But as a starting point, we might follow what the EU does, which is to take the approach that any new compound or approach should be no worse than the ones currently in use. So if we think about that specifically for our toxins, the anticoagulants and 1080 are the ones that are used more commonly. And if you have a look at this figure on the side here, this is a summary of some work that we did about 15 years ago, looking at the relative welfare impacts of different toxins used in New Zealand. So what I want you to note here is on the right-hand side, you'll see we've got brodifacoum and pindone in the red circle there. These were judged to have the most extreme and long-lasting impacts on the welfare of different mammals. And so as a starting point, we might say that any new toxin or refined toxin that we develop should be no worse than the anticoagulants and preferably much better. The second question is, how can we build animal welfare into our development pathways at every step. And I think a key principle here is that we want to develop these pathways to fail fast. In other words, we want to stop the development process early if poor welfare outcomes are predicted. This is really important because a great amount of time and money can be invested in the development of novel products or new uses of products. And if people work all the way through towards the end of the process and get towards authorization or registration, there's a really big investment in that going all the way through to the market. And we want to make sure that animal welfare actually gets the consideration that it deserves and that it doesn't get overridden at that point because so much investment has been made. So what we're trying to do right now is we're trying to essentially map these pathways as they currently exist. We're trying to understand how and when animal welfare is considered in pathways to new product registration or registration of new uses. And we'll start by doing that in the New Zealand and Australian context, but I'd be really interested to hear from people involved in this kind of process in other locations to find out how it might be the same or different. There are a whole bunch of different questions we would like to answer in this mapping exercise. So, for example, what kind of research is required that might involve animals? What's the timing of different kinds of research on aspects such as the efficacy of the product, what any non-target effects are, so which other non-target animals might be sensitive to this compound or molecule, and what impacts are there on the target animal in terms of welfare. We're also interested to know how welfare is assessed, so what kind of system is used. Are the outcomes comparable across different research groups, for example? And who is doing that research? Do we have a structured approach to welfare assessment where everybody can understand the outcomes? And do we have the appropriate degree of independence in that testing? We also want to know how the 3 Rs are applied to reduce harms during toxin development and how the welfare outcomes from any research that's undertaken are used to inform or modify the next steps in the registration pathway. Coming back to the third question, how can we know or predict how harmful a new toxin is likely to be? The approach to predicting welfare impacts, and remember what we really want to do is fail fast, I think will depend on what we know about the toxin. So it will depend on what we know about its mechanism of action. So for example, its molecular targets and its action at cell and tissue level. It will depend on what we know about the pathophysiology, so what actually happens in the animal's body, what are the effects on organs and the whole animal. And what do we know about the physiology and the behavior that's specific to that target species or taxon? If we think about refinement pathways, we probably know quite a bit about the toxin's mechanism of action. And so what we want to do is bring that information together and develop an understanding of the species-specific factors that are likely to influence the welfare impacts for that particular target. So we might want to know things like the target animal's receptor distributions or densities that are relevant to the pathophysiology. When we think about novel toxins, this becomes even more challenging, and usually what we need is more fundamental information. So this is where we've got some really great opportunities for systematic and robust 3 Rs applications to reduce the impacts of the research that we would do throughout the development pathway, because animals will almost invariably be used during the development of these new toxins. So a couple of things we've been thinking about. The first is to develop high throughput models that don't use any animals to give us that information we need about the mechanism of action and the molecular targets. And one of my collaborators, Clive Marks, has been working on a fungal model to develop a better understanding, of the mechanism of action of 1080, which has not been very well understood to this point. Another example might be to reduce lethal animal assays with less harmful sublethal assays. So we might want to use early predictive indicators of lethality so we can have much earlier humane endpoints in our research, or we might want to look for lowest observable adverse effects, for example. So just a quick summing up before I finish. I think it's safe to say that lethal control of pest animals using toxins will continue, but there's an appetite for new or refined toxins that are less dangerous to our valued non-target animals, but also remember that we're aiming to have those be less harmful to the target animals' welfare as well. I think we can do this by building animal welfare science into every component of research, registration, and application of these new or refined toxins. And we might do that by including animal welfare scientists into our teams right from the very start, by applying structured and science-informed approaches to animal welfare assessment, by replacing animal use or reducing animal harm in the process of toxin development and applying the 3 Rs in a really systematic way at every step, and aiming to fail early so that we reduce the overall impacts of toxin development pathways on animal welfare. As I said, we're really early in our exploration, and I'm very keen to hear ideas from people who are participating today. And I'd just like to finish up by saying thank you so much to my collaborators in New Zealand and Australia. Thanks so much for the FSVO and UFAW for supporting me to be at this conference, and to our funders. And I'd love to hear from you. Thank you.
Thank you. So obviously, sadly, we can't ask Nio any questions, but we'll try and raise some of her points in the discussion later. But for now, I'm just going to pass straight on to the next talk, who thankfully is in the room. So welcome to Andri Manser from the University of Zurich, who's going to be talking about using naturally occurring sterility genes to humanely control house mice.
Thank you very much, and thank you very much for the organizers for giving me the opportunity to speak here. We've heard a lot about this morning about house mice as laboratory animals, but I want to highlight in my talk that they also exist in the wild, and actually they're a massive problem as an ecosystem pest. So quite in general, we've heard before that actually invasive species are a main contributor to our biodiversity crisis we're actually facing on this planet at the moment. And it actually has been estimated that about 50% of animal extinctions are caused by invasive species. At the same time, a lot of our ecosystem diversity is actually on islands. So 20% of animal diversity occurs on islands, despite only being 5% of the Earth's surface. And house mice have followed humans around the planet. So we've conquered the planet, and they've sort of followed us. And they have been identified as a key problem on almost 60% of islands. We've heard in the 2 previous talks that there has been a lot of attempts to get control of this problem. So about 120 eradication attempts have been happening since the 1950s. And we've also heard that the current control practices are far from ideal. So it's these anticoagulant poisons that kill the animals very slowly. So that has raised huge animal welfare concerns. It poses a risk to human health if it goes into the water. And also we've heard about off-target effects, so it might kill other animals that are in the target area. I want to take you to a particular place in the South Atlantic called Goth Island, which is a particularly stark example of what house mice can do. So this island is in the South Atlantic and is home to this critically endangered Tristan albatross that exclusively breeds on this island. House mice are thought to have been introduced there in the 19th century by seafarers. They've almost doubled in size. So, and they've started to actually attack bird eggs and even live chicks, leading to a massive eradication attempt. So there was a huge endeavor in the summer of 2021. Where they actually used this coagulant poison approach that was broadcast with helicopters. This was actually quite a success, so people looked at the breeding success over the years, and as you can see, in the year following the eradication attempt, breeding success has skyrocketed compared to before. But already in December 2021, there were first signs of, or first house mice were detected again. So it seems like that this 10 million program was successful for a while, but actually it's really hard to control, and it seems like that the mice are back and probably on the rise again on the island. And just to highlight that mice are not only an ecosystem problem, but they're also a huge agricultural problem. Here are some crazy images of 2021 in Australia, where they have these massive outbreaks, irregular outbreaks of house mice, and it really looks like a biblical plague. Yeah. So what we're doing in our research group in the University of Zurich is to look at a sterility gene called sterile T, or simply T. This gene has evolved in the house mouse cluster about 3 million years ago, so it occurs naturally in house mice. And it's actually not just a single gene, but an entire, almost a third of a chromosome. So it's on mouse chromosome 17. It has two characteristics that we think could be interesting for pest control. So as the name implies, males that carry the T, so are homozygous for this particular genetic entity, are completely sterile. So if a female mates with a mouse, with a TT mouse, so all this sperm is rendered dysfunctional, she will not produce any offspring. So akin to sterile insect techniques, this could sort of be used if you release sterile animals into a population to sort of transiently maybe drop the birth rates in a population. But obviously that wouldn't work in the long run, because there's strong selection against being sterile, and as soon as these animals die, sort of this effect would disappear. Interestingly, this gene has a second twist, namely that heterozygote animals are not only completely fertile, but they actually transmit it not only to 50%, which is the normal expectation under Mendelian inheritance, but they actually manipulate sperm such that only the T is transmitted to the offspring, or in 90% of the cases. This is an effect called gene drive, and the release of sort of heterozygote individuals could then, from an evolutionary perspective, be used to release animals into a population, and it will actually propagate genetically despite the negative cost it has for the individuals in terms of the sterility effect. So that could suppress populations permanently, not transiently. That's a typo there, at least in theory. We wanted to figure out whether this could actually work or not, so we built a little model where we imagine an island of resident house mice, so that would be a pest population, and then the idea would be to bring in and release either sterile or heterozygote animals onto the island. So the model has these different two release rates. Then these released mice would need to be able to settle in the population and also establish dominance territories on this island, which our model tries to take to account for. Animals then need to survive, so there's a survival, different survival rates, although we have these animals in the lab, and there's no indication that there's any survival differences between these T carriers and wild-type animals. And obviously, the main goal of this would be that they need to attract mating partners, which then would lead to this effect that females mate in vain, and that would cause birth delays. I want to elaborate a little bit on the exact reproductive cost that this could impose. So if you imagine a population with no sterile animals, typically it takes a certain time for a female to find a partner, to mate successfully, to raise her young, and then to be ready again for the next reproduction. So this gray arrow there would be sort of a typical interbirth interval in a population without any steriles. So in this case, if you look at the standard amount of time, she would produce, say, 4 liters. Now, if we add some steriles into the population, she may first, again, encounter or mate with a normal resident male, produce offspring, but then if she encounters a sterile male, that would obviously then maybe cause a small birth delay, and sort of maybe an Easter cycle or 2 later, she might be able again to encounter a wild-type animal. So the idea there is that these delays would cause a slight decrease in the total reproduction. And obviously, if you have many steriles in the population, this effect will get stronger and stronger to a point where everyone is sterile, then obviously that will go to 0. that's one option that she sort of, that's our goal, would be our goal, that she mates with TT steriles, but obviously she could also mate with these heterozygotes, in which case you would create more steriles as offspring, and then they could sort of genetically propagate in the population. So these are the two mechanisms by which this could work. I'm now going to show a brief sort of what we think, based on the current knowledge, what a scenario could look like. So we have here on the y-axis time. So at time point 0, we would release certain animals into a population. In blue, we have a resident population. So here you have mouse density. So the blue shade is the density of mice, resident mice. And this dotted line here would be the frequency of steriles. So if we let this play out and we release heterozygote animals once into the population, this is what the dynamics would look, at least in theory. So you see that thanks to this gene drive effect, you would have a rapid spread of these sterile animals in the population. And that would then cause a slight decrease in population sizes. In this case, that wouldn't be sufficient to completely eradicate a population. We can then obviously theoretically ramp up release efforts. So we could, for example, combine a release of heterozygote animals with these direct releases of sterile animals at 5%, which then would cause a further decrease in population size. Still not sufficient for eradication. If we do that sufficiently, then we would end up with a scenario where actually the population goes completely extinct. And so for each of these parameter scenarios, we can sort of calculate the critical release threshold that would be required to successfully eradicate the target population. And we've done that for a number of our model parameters. So here you see an example of the minimal required release effort as a function of this delay penalty that would be caused by mating with these sterile animals. You see sort of the minimal release required for eradication. And as you would expect, you need to release more animals if the penalty gets smaller and smaller. And these crossed out areas here would be parameter regimes where this wouldn't work at all. We've done this for a number of the other parameters that we looked at, for example, the attractiveness of steriles, multiple mating rates, gene drive strength, and so on. And what we find is that this method could work in some cases, but it's really parameter dependent. And in some cases, it's really important to know where exactly we are here on the spectrum. And that's one of the problems. We will know so far very little. We have little information about all these parameters. And I'm part of a wonderful research team in Zurich, led by Anna Lindholm, where we actually try to fill these gaps by actually measuring some of these model parameters to assess the feasibility of this method. So we have 2 PhD students. So Salome Freire is planning to do laboratory experiments where she wants to, in detail, analyze the effect of the sterile on reproduction, whether females actually would even distinguish between steriles and fertile males, whether they recognize them. And she also wants to measure the strength of gene drive in our population. And then Kaspar Gödecker, who is another PhD student, wants to do enclosure experiment where he wants to figure out whether mice can actually settle into and establish in an existing population, and also measure the impact of these steriles on reproductive rates. So really parameterizing the model in that sense. We also study a field population of house mice for over 20 years, so it's quite a long-term study. And one of our plans is to actually, so Kaspar will start to release TT sterile males into this population to see whether that actually could work in an even more natural context. We think that this method, if it would actually work, as I showed you before, the model shows that it could or it could not, depending on what we find, would have some outstanding benefits, we think, to over current control methods. So first of all, it would be completely humane. So animals are not really killed, but they would just not be able to reproduce at sufficient rates to maintain populations. It would be safe in the sense that the T sterile is completely specific to house mice, and it sort of wouldn't mean any poison in the environment. It would be quite simple because we already have T sterile mice in the lab and in the wild, and they could be quite quickly bred on local backgrounds or the background, the genetic background of a target population. So it could be breeding these animals could be relatively cheap and straightforward. And finally, you may have heard that a lot of these gene drive can now also be produced synthetically. So T-CRISPR or CRISPR has been very successful, and gene editing has brought us to a point where we actually can design these in the lab. And we think that actually this being a natural variant of a selfish gene or a gene drive may make this much more easy to use in a context, in natural populations. And with this, I want to thank the wonderful team in Zurich. I want to thank our funding. So our project is funded by the Swiss National Science Foundation, and obviously you also for your attention. Thank you.
Thank you very much, Andrew. Since you're the only panel member, why don't you have a seat for your questions?
A one-person panel.
Yeah, sorry. I'll join you in a minute. So first of all, I'll take if there are any specific questions on that talk, and then maybe we can get into some of the more general issues. So raise your hands if anyone's got questions in the room, or if there are any online for Andrew in particular. There's one over there with you guys at the back.
With falling mouse population, presumably there will be less competition for food. What impact would this have on litter size? Might you expect increased litter size, reduced infant mortality, and increased survival to adulthood? If so, have you included this in your modelling?
Yeah, that's a very good question. So yeah, the model at the moment has sort of density-dependent birth and death rates. So birth rates go down with density and death rates go up with density. And then you end up with this sort of carrying capacity where birth and death rates are in balance. So that's currently in the model. But there's obviously a lot of interesting things that could happen as you get to smaller and smaller densities. For example, one of the parameters we have in there is remating rates, which is something that might change. And we've actually done a lot of research showing that that's a really important predictor of how well these teeth can spread, because these T animals are actually, these T males are sort of compromised in their sperm competitive ability. So whether this works or not is quite strongly dependent on the remating rates in a population. And that's, for example, also something that could change with population density. So we certainly need to find out more about general mouse demography. So how do birth rates change with density, and also death rates. So this is also turns out to be a really important question in terms of how this could work or not. Yeah. Thank you.
Could you just wait for the mic, please, and say who you are, please?
I'm Andrea Vieira. I'm an animal welfare scientist from Brazil. In terms of the welfare of the mice, would you be introducing, would the gene introduce any disease for this animal as well? Did you, have any assessment of that, so that that could also bring diseases to people, right? Depending on the immune status of the animal.
I mean, in terms of, I guess you would, if you would want to use that on a target population, you would probably do back crosses onto that local strain, so that the only difference between the population that is there, the sort of the target population, would be the specific gene that you're using. And I don't think we have any evidence that that is the gene itself is associated with any disease.
Microphone. Can
Yeah.
you use the microphone, please? I had a question, which was, assuming that this effect isn't powerful enough to eradicate on its own, could it be combined with the classical low welfare rodenticide approach, perhaps?
Yes, we thought about this, and also because they're sterile, we've also wondered, because then they wouldn't interbreed with the local population, how you could make the males even more attractive. Sort of, we call it Adonis mouse. So if we could design a mouse that is really attractive, because they're sterile, there's not a danger that we leave behind a more attractive sort of mouse population. So we could play around there. So there's other ways, sort of, if it's combined with trapping or if it's combined with something that makes them more attractive. I think we currently don't really know what mice are really attracted to, but that would be different ways of actually making this more effective if one would combine that. Yeah, and there's also a group in Australia that actually plans to use this and then uses CRISPR that targets as a fertility gene. So there's also plans afoot to use this in combination with some gene modification to actually make it a more powerful mechanism genetically.
Interesting.
Yeah.
And are the Australian authorities happy to proceed with this? Do they have any safety concerns or other issues?
So a small disclaimer, I'm more of an evolutionary biologist or a behavioral ecologist, so in terms of pest control, I'm not quite sure. And I mean, what we are doing is fairly early sort of baseline research, trying to figure out whether this could work in principle, and I guess we're sort of far away. But the impression I get is that they're quite keen on getting rid of these animals at almost any cost, is this is the impression I get at least.
I'd be interested if there's any regulators in the room who would comment on their willingness to license it. Yeah. But I saw Leslie had a question over there, if we could get the mic across.
Thanks. This may sound a bit naive, but they say that everything plays a role. Now, I know they're invasive species on some islands, but not Switzerland. Is there anything negative that could come out of this if we completely eradicate mice in certain countries or islands, and then one comes on my boat and I bring it to Switzerland and now the owls don't have anything to eat?
Yeah, I think at the moment we're more worried about it not working at all than actually being so successful that you might accidentally kill all the mice on the planet. And I think ethically, as you alluded to, I think it's quite, at least to me, it's quite an important distinction that we are, in these places, we're trying to undo harm that we've already sort of caused in the first place. So I think there's a sort of a distinction between places where these animals occur naturally, maybe ethically, and places where we actually try to save the local populations of other animals from the harm that we've sort of imposed on them.
Excellent. Is that a hand? Yeah.
Hi, thank you so much. I really enjoyed that. In certain species where that have been given contraceptives, so wild horses, for example, or kangaroos, what they found is that often the females will leave the group and try and join another group. And it's thought to be because of the low birth rates. Essentially, the females aren't getting pregnant and go, this dude's a dud, I'm going to go find someone else.
Yes.
Do you think that might be a risk with your population, and you may inadvertently end up with a whole bunch of migrating mice moving into new areas?
Yeah, that's really interesting. Actually, we've done some research into just a separate question. We had some other evolutionary reasons to think that this gene might actually affect the migration, but that's more than and we've actually found systematic migration propensity differences between mice that carry this T haplotype and mice that don't. And I think also how the females actually would react is a really important question. One would think that there is mechanism for them to detect sterile animals, sterile males, and if they don't get pregnant for a while, that they would sort of try to mitigate against that. So that's what Salome is exactly trying to do, sort of trying to do mate preference tests, and also in the enclosure we're hoping to see whether that works or not, and how that, But that's certainly something that's really important to know.
Beautiful, thank you so much.
So I guess what you're implying there is that this may well work much better on a very small island than at a continental level, which might actually be quite a good thing if we're concerned about eradicating the house mouse.
Yes, that's a good point. Yeah.
Indigenous territory. Have we got any questions online over there? Oh, we've got a real world one at the back there, further back. Oh, Hans.
Thank you very much for your interesting talk. I was wondering, have you considered whether this technique can be used for invasive species in inland waters? For instance, an American freshwater lobster is really an ecological disaster in the Netherlands, in rivers. So it's...
Yeah. So these gene so one of the things that I think that makes this quite powerful, that is very specific to house mice, is also one of its problems, because this gene only occurs in house mice and probably only works in this specific species. So the fact that it sort of occurs naturally also makes it sort of limited in terms of how it could be applied to other species, at least. And I guess it's no accident that we find these gene drives in species that have been studied very extensively, that have been sort of genetic standard or genetic organisms. So we found them in many places, and it would probably be interesting to know whether there's, wherever we look, basically we find genes like this. Whether there will be others out there, but I think our knowledge of that is very limited, how they're spread. But at the moment, this specific approach, I think, would only work specifically with house mice because it has evolved in that species. Yeah.
Thanks. Was there another question at the back there that I saw? Oh, there's one on the other side, just at the back. Josh.
Hi, I'm Josh from Vet Record. I just wanted to ask about Gough Island. So did they successfully eradicate all the mice on Gough Island and then somehow found their way back? And if so, how? Or did they not manage to kill all of them and then the ones that were left reproduced? And if it was the former, presumably you wouldn't have the same problem with them eating the chicks because they wouldn't have evolved to be big yet if they're new ones being reintroduced. I mean, what was the mistake that allowed them to come back?
Yeah.
If you like?
I'm not 100% sure, but the impression I got was that they just didn't manage to eradicate them completely. So, and obviously reproduction rates, as we heard earlier, might be really fast if there's only a few because there's not much competition going on. So they may actually bounce back quite quickly. So something we find in our natural population, that if population sizes go down, it can be incredibly quick. And I guess that's also one of the limitations with these poison approaches, that you really need to be able to get rid of all of them in order to permanently actually solve the problem, because otherwise quite quickly you may actually be back to square one. Yeah.
Yeah, and that's clearly a welfare worst case scenario, I guess, that we just end up constantly cycling through, killing vast numbers of animals on an island. There's a question at the front, Jess.
Jess Martin, Newcastle University. Thank you very much for both of your talks. This one's maybe a bit more towards you, Hugh.
Oh, no.
So Naya made a really good point in her talk that, the method that the EU has at the moment for allowing new methods is whether or not they are the same as what we currently have, or they are better, so they can't be worse. From an animal welfare point of view, do you think that bar is high enough? And given what you said in your talk, that there's so many knowledge gaps of the methods that are currently allowed, how easy is it to compare to those new methods that come in?
It's not easy to compare. I mean, that's genuinely true. I mean, I think the amount of research that gets done on actually understanding the impacts of rodent control is quite small, because who's going to fund it? Research gets funded to develop new methods by the industry because there's money to be made, and that's fine. But I think, in terms of funding the research, we don't have the funders who would fund lab animal work or possibly even farm animal welfare. So that is certainly a problem. Setting the bar is a very tricky one because I think when I started out talking about this years ago, made some quite sort of glib comments about, that we shouldn't do certain things because they're incredibly bad for animal welfare. But, these are genuinely issues that threaten human health and global food security. You can't just say, I'm going to abandon rodenticides in Africa because the food system will collapse until you have something better to do. So we have to think very long and hard about this. And that's why I also mentioned efficacy in the research that's needed, because we need to know what works as well as what's humane, because it's going to be really dangerous to ban these methods. Some things we can do. I mean, glue traps in the UK was strongly debated, but I think, I genuinely believe we can cope without glue traps in the UK, and I guess we'll find out now we have a ban, and other countries have already done it and don't appear to be completely overrun with rats and mice. So cautiously there are things we can do, but I think we are going to have to live with some very unideal methods for a while. In terms of the approach, I can't see a better approach than let's try and develop something that's better. But I guess someone, ideally the regulators amongst others, needs to fund the research to check that, and that's, you know, incumbent on them.
I'll follow up with then really quickly is, so for everybody in the room, is how do we get attention to increase funding for this area? Because it is really important and it's not got that attention.
Good point. Well, one thing, if we're not talking about the regulators, we talk about the numbers and we talk about the impacts and persuade those funders who do care about numbers of animals and care about a scientific approach. And I think we know some of them who've traditionally focused very much on issues in livestock welfare because they're motivated by a straightforward sort of consequentialist, largest impact on the largest numbers of animals. And I think we can make a pretty convincing case that we're talking about many, many millions of animals with a very significant degree of suffering. So I would hope someone might step in that field. I won't name any names. There's one over there.
Thank you. I just want to give a little bit of anecdotal story about rat eradication. I am a vet, but I also am an organic pig farmer. And I had a rat problem on my farm, which in the end I am not allowed as an organic farmer to use any rodenticides. So I started calling around in the Netherlands and I found a person, a pest certified pest controller, and he started shooting the rats. And that was very successful. I really have to say it is pretty basic, but a lead injection really works. And he, in the end, he shot around 400 rats, which is a lot, but I have to explain. I feed my pigs also with all the leftovers of the only organic supermarket chain there is in the Netherlands. So, and as known by science, rats prefer organic above conventional. Yeah, they did through research on that. So, and then after a certain period of time, his 1 hour or 2 hour shooting times in the evening were only bringing 1 or 2 rats. And then we said, okay, let's now go back to conventional control system using wild animal. And we brought in a rescued polecat. I hope that's the right English word. And that polecat is now controlling my farm. And the pest controller, came to my farm last week, and he saw with his warmth camera, he saw 1 rat. That's it. So just by using the basic stuff, you can come quite far. But in the end, it was a pretty high bill I had to pay because he shot many hours. But it worked, and there is no pesticides used, and everybody happy.
Okay. And I mean, it does emphasize, I think, at an even more basic level, before we resort to these methods, particularly when we talk about rats and mice in buildings, stop them getting into the building.
Yep.
Is the absolutely fundamental thing, rather than fighting this constant battle. It doesn't solve the problem with invasive species or probably the threats to crops in Africa, but certainly for the sort of domestic scenario, it really does form the first line of defense. How are we doing online? Excellent.
Back to Andrew. There have been experiences releasing infertile males in insect populations. And so have you studied their experiences?
Yeah, they've actually been quite encouraging and super successful. I guess the critical question is how much can these approaches be translated to... so I think they've been some of the most successful pest control efforts that we've done. But obviously there you can release millions and millions of, say, mosquitoes at one point in time. And the question is, how is that translatable to rodents? I guess you couldn't just flood a population with, I don't know, 3-4 times the resident population size. That would, obviously they would live for quite a while, so transiently you would increase the impact that has on the local island much. So I guess there is a little bit of a question mark as to how translatable that is to vertebrate species. Yeah.
Okay, thanks. If there are no hands up in the room, I was just going to ask if anyone had any questions or comments on Nio's talk. We can relay some of those back if you do. I thought that was a really interesting approach in New Zealand, and it is really interesting to look at how complicated it's going to be to develop these solutions. And the analogy that, again, I had drawn to me was it's like drug development. You know, a huge amount of effort is going to be invested in things that don't work, because that's the nature of doing science, and it's going to be fundamental that we get the basic science right that predicts what compounds will work in nature, so that we don't make the mistakes that the pharmaceutical industry used to make before they realized they were in the middle of a replication crisis. But it's, again, it's a point that I had made to me early on when we started talking about this, is that people really are working really hard to develop solutions to this, but most of it is not successful because it's a really hard problem. Did anyone have any comments on that approach? I can see a hand right at the very back corner.
So for the online speaker, and given the two talks together, I'm wondering if instead of looking at more toxins and poisons in New Zealand, there could be an approach with a pharmacological fertility control. I mean, would that be possible?
I've certainly heard it discussed, and I think for some of those rodent species, it's a plausible approach. There are some compounds used in the US, I believe, so we have some people actually using fertility control, if I'm right. Yeah. So I guess, again, it's the concern about releasing those compounds and whether they have accidental non-target effects, particularly in New Zealand, where they're so keen to protect their indigenous wildlife. Well, I think we're almost perfectly at our lunch break now. So first of all, we should thank Andri for answering a vast amount of questions. Thank you very much. And Nio for fighting her way through sickness and COVID to record her talk online. So thank you to both the speakers. It's lunch break now, which is, well, as you come out of this door, it's over at the other side of the building and slightly up in that direction. You should have all got a little card or ticket, which is your free lunch voucher, which you should leave on the table in the canteen, and then you don't need to pay for lunch. So enjoy your lunch and come back at, is it 2:30? I'm just checking. 2:30, yeah. So have an excellent lunch, everyone. Thank you very much.
Okay, everyone, please take a seat. It's a pleasure for me to start the session in the afternoon. We are again speaking about slaughter of livestock. Before I go deeper into the topic, I was also asked to present myself a little bit, because not everyone knows me. Also, I'm a veterinarian, to start with that. Second, I have been for 18 years at the University of Zurich at the Institute for Food Safety and Hygiene. Then, about 5 years ago, I changed to the FSVO and has been there for 5 years. What I have done, basically, I'm not coming primarily from the animal welfare side. Almost my whole life, or the professional life, I must say, was really that I worked with microbiological hazard along the food chain. This means I had a lot of little bacteria that I looked how resistant are they, what are the pathological factors they have, and so on. After changing to the FSVO, it started really intensive that also animal welfare at slaughter was of importance. And when I remember, Martin Rice said it in the beginning of this day, 30 years ago, during my studies, I didn't hear anything about animal welfare at slaughter. And today, I have to say, as a society, they are really adapted. They want to know what's going on. Not all the people, I also have to say, especially when you come with a topic like slaughter, then it's not so that anyone likes that. I was also asked before, do I still eat meat? Yes, I eat meat. But I know that the animal had to die for that. And for me, also when I had this with the students, was always important, everyone can eat meat, but please be aware what happens. The steak starts not in the refrigerator. So, and now, closer to the topic itself. Also, I have already said, slaughter. This means meat for human consumption. And with that, I can also already say, not all methods that you could use are also allowed to use. Because when you want for human consumption, then you have a little bit other framing condition. We speak about killing. In the terms of slaughter, killing is a two-step process. It's stunning, and then it's bleeding. Stunning means unconsciousness. Bleeding means dying. These two steps, when everything goes right, then we are all happy. But exactly when we speak about slaughter in a narrower frame, then it's really the stunning and bleeding, and especially the stunning, of course. And the stunning methods that are allowed for each species, they are also defined in the legislation for the normal red meat animals as we know them. This means that it's defined what methods you can use or cannot use. What I have to say is, when you take an example of pigs. Pigs, there are slaughtered in Switzerland 2.5 million each year. When we go to Germany, it's over 60 million. We have, in a modern slaughterhouse, it is normal that 250 pigs are slaughtered per hour. When you go to poultry, you can go up until 15,000 per hour, and this only with one slaughter line. If you have big pig abattoirs with several slaughter lines, you can increase this. And now, I'm not making a judgment if this is good or it is not good, but it's at the moment reality. And when we really want to look for the animals, to protect the animal welfare, then it's important, what can we do? And what can we do that is also still profitable for the others? That's the reality. And for the amount of meat we need, it's at the moment not possible that each single animal is slaughtered at the farm, because to this amount you will not get. You will never be able to do this. What I have to say, I will not go into the different methods for stunning, but only an example. When we take for pigs, electrical stunning, the stunning normally works. I speak of a standard fattening pig that goes through a restrainer. The problem there lies otherwise, because how can you get the pigs in a single file? On the other hand, if you take carbon dioxide, then you can put several pigs together, they have companion. It is normally easy to operate, but I think we have heard enough about all the people, what they have told about adverse effects. I will not start with that. But so you see, we have disadvantages and advantages with all of this. What I also have to say when I speak of animal welfare at slaughter, it's not only the stunning and the bleeding. It starts a long way before. Because as long as the animal is alive, this means the transport, this means the holding, and so on. A lot of people concentrate absolutely stunning and forget all the rest. Please do not do that. Okay, I'm very pleased that we have our own session for this topic. We had the last 2 symposia. We were lucky to get almost an own session. We have a lot of different talks that are coming up. They are really various. We are starting about the European Union Reference Center for Animal Welfare, and there with the specific focus on rabbit and poultry. Then we will go to indicator and sensor technologies with pig welfare. Then we will have a talk about alternatives to carbon dioxide with these 8 gas mixtures, or the speaker will tell us more about this. And finally, we will also cover shortly electrical stunning. And now you see also why I have selected the pig as example for here. But please do also not forget, it's not only about pigs. And that's why I'm really happy about the talk about poultry and rabbits. So this would be my very short introduction. I think we can move on then directly to the first speaker. The first talk will be presented by Alexandra Contreras-Yoda. She will speak about the role of even this mentioned reference center in refining the assessment of poultry and rabbit welfare at slaughter. She comes from the Institute of Agri-Food Research and Technology in Spain. The floor is yours. Thanks a lot.
Thank you, Claudio. Good afternoon. Thank you to the organizers for giving me the opportunity to expose the work we've done in the context of the European Reference Center for Animal Welfare for poultry and other small farm animals. Today, I would like to present the role that we have in refining the assessment of poultry and rabbits' welfare at slaughter. So the EURCAWs were born in the context of the regulation of artificial controls and are intended to help at implement the legislation to competent authorities and member states of the European Union. There are 3 of them. The first one was the EURCAW pigs, then the EURCAW poultry and other small farm animals. Other small farm animals mean minks or rabbits, for example. And then we have the EURCAW ruminants and equines, and soon will be the EURCAW fish. In the context of the EURCAW poultry and other small farm animals, we are 4 institutes of research. It's ANSES, led by Virginie Michel, IRTA, led by Antonio Velarde, ISLER, led by Leonardo Binco, and Aarhus University, led by Anya River. One of the priority areas is the stunning and killing, which is why I'm here, to expose this. This is the slaughter team, Virginie, Antonio, Arantxa, and myself. And the topics that we address is the water bath stunning in broiler chickens and turkeys, the gas stunning in broiler chickens, and the electrical head-only stunning in rabbits. Why these methods and these species? Because although there are several methods to stun these animals, these are the ones that are more used. In fact, 50% of broilers are stunned with water bath and half gas stunning. And in case of rabbits, 99% are stunned with electrical head-only stunning. Seeing this, the main role that we've done in the last 4 years is, first of all, do reviews, also meetings with CA, a couple of meetings per year, and also we send service to competent authorities and official veterinarians of all along the European Union. When we identify gaps of knowledge, we try to make scientific studies, and with these scientific studies, we try to create knowledge, and with the purpose of disseminating this in form of reports destinated to official veterinarians, which is not so scientific, more at at a level that everybody can understand. Also, webinars, fact sheets, and videos. So first of all, in 2020, what we did is to take the regulation 1099 of 2009. We took this regulation and we broke down into small pieces of regulations. And we select those are related to the state of consciousness. And with this, we try to make a list of animal-based indicators, resource-based indicators, and management-based indicators to make the assessment of each legal requirement. And then we wrote a method for the assessment of compliance. All of this using guidelines and materials that are currently at disposal. What we found is that 16 legal requirements linked to the assessment of the state of consciousness, only 5 of them can be evaluated using ABIS. So this is more resource and management oriented currently. The gaps of knowledge we identified is that there's no validation of animal-based indicators on field. We know that the EFSA did a great job in 2013 and 2020 to make a list of valid and feasible indicators, but the repeatability and the prevalence of these indicators were not addressed. So what we've done is to visit 5 commercial European Union slaughterhouses, 3 in France, 2 in Spain, with the objective to refine this list that EFSA provided based on feasibility, inter-observer repeatability, the prevalence of these indicators, and the correlation between indicators. When I say prevalence, it's not the same as we found that vocalization is hardly ever heard, and breathing is most common, so why don't you put attention on breathing and not to vocalization? It's an example. Then refine the method for assessment, and also report the efficiency of stunning according to key electrical parameters. The results were published in Animals, and also there's a report that is for the official veterinarians in our webpage, which I will show you later on. We did the same in turkeys because EFSA provided the list of indicators for poultry, but it's not species specific. That's why we visited 8 commercial European Union slaughterhouses in Spain and France as well, and we did the same objectives: refine the list based on feasibility, blah, 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 refine methods, and report efficiency of stunning. And this, all of this was also published, but also is reported in our webpage. For making it more handy, we create fact sheets. These fact sheets have this information summarized, and also we have it in several languages. English, Spanish, French, Italian, and German. And what we have here is a small introduction, the main indicators that we have to focus on, the slaughterhouse conditions. Also, we have QR codes to check for videos and how to assess these indicators. And also, on the reverse of the page, we have the sample size calculation, which is also that it was difficult for the official veterinarians to address. Same for the turkeys, with the QRs, most similar. We also have some videos, motion videos, which are also published, are freely available. And we found a good practice in a turkey slaughterhouse that they were restunning the animals using a captive vault for sheep, but it was good. So we report it here, and also with the QR code, we have a video on how we see it's performed. Additional considerations, this is not part of our objectives, but it's something that I want to highlight in this conference, is that for broiler chickens, runs are at high risk of skipping the water bath or the neck cutter. It's something that we've seen more times than we were expecting. Also, pre-stun shocks in almost all turkeys. If you know that the turkeys have the wings that hang below the head, and you can, if you pay attention, you can listen them vocalizing at the entrance of the water bath. So I think it's still a non-addressed issue, although in the HSA webpage they have a way to prevent it, but at commercial level it's not performed yet. And also we found in broiler chickens that when the slaughterhouses choose to put the frequency above 550 and 600 Hz, there is a high prevalence of conscious broilers. I think it's not the induction, it's the length of the unconsciousness is so short, so they regain consciousness before dying. Another thing is sexual dimorphism has to be taken into account, especially, of course, in turkeys, because sometimes you know that males and females are raised separately, but sometimes there are mistakes. So when you find males in a batch of females, these males are not well stunned because these electrical parameters are not designed for males, which are bigger. And when it's the opposite, when we find females in a batch of males, these females skip the water bath. So they appeared fully conscious at the moment of neck cutting. So, of course, this should be taken into account at the slaughterhouse level, at least separating in different batches and stun the animals according to the size. Also, the electrical parameters should be adapted according to average body weight of the flock. It's not the same stunning animals of 4 kilos that stunning with the same parameters animals of 20 kilos or 16 kilos. So the efficiency varies. So, key message, do not rely only on electrical parameters allowed by legislation, since do not guarantee efficiency stunning in all animals. Sometimes there are a percentage that regain consciousness. And because of this, it is of utmost importance to check the efficiency of stunning using the recommended method and animal-based indicators. On the other hand, as I said, that the 50% of the broilers in the European Union are stunned using gas. So we performed a review on the main welfare aspects of stunning broiler chickens by exposure to controlled atmosphere. The methodology was, again, send surveys to competent authorities and official veterinarians, do some scientific literature research. And the content of this report that it's intended for official veterinarians is to describe the list of controlled atmosphere stunning methods that are allowed by legislation and which of them are in use, because there are 5 methods, but only 2 are currently in use. 2 and a small percentage, maybe 3. And for each stunning methods, we put the legal requirements, also the available equipment, the positive and negative welfare aspects. And with all of this, we have conclusions, and we also have highlight the gaps of knowledge. We have also a thematic fact sheet in English and the other languages I described before, with the positive and negatives and something that can be handy and useful for the inspectors. With the gaps of knowledge, we performed a scientific study at EURCAW, the institute I work, because we have experimental slaughterhouse. We tested 3 different gas treatments. The CO2 into phases, that is the most used. I think 99% of the slaughterhouses that use gas method use CO2 into phases. And we tested 2 alternatives to CO2 into phases, which is 40% CO2 and 60% nitrogen, and 20% CO2 and 60% nitrogen. The objectives were to assess the onset and duration of unconsciousness and time of death, and also characterize the gas aversive response by correlating or making a relationship between EEGs and the behavioral indicators. And with this, discern which gas treatments improve chickens' welfare. And all of these objectives were accomplished, and we have this also published as Ruthink et al. 2024. We also gave some webinars to official veterinarians. This is the one about water bath in chickens, water bath in turkeys, and water bath and gas in broilers. Also, in the context of rabbits, we started with a review, the main welfare aspects of the different stunning methods in rabbits. We send surveys, we make literature review, and we put the list of stunning methods, which are 4 or 5 as well, but only 2 of them are used, which is 99% head only electrical stunning and captive bolt, 1%. So for each stunning method used, we describe the key parameters that are currently in use in the European Union, the assessment of the state of consciousness and death, and the potential risk for rabbits' welfare and good practices. And with the conclusions, we added the gaps of knowledge that we found. And with these gaps of knowledge, we went to 15, this time was 15, slaughterhouses in the European Union, 5 in Spain, 5 in France, 5 in Italy, with the objective of refining the list of the indicators reported by EFSA in this year, based on feasibility, repeatability, prevalence, and correlation between indicators, in order to refine the method and report the efficiency of stunning of different electrical parameters. This report will be finished in September and probably will be available online on October. We also, for the official veterinarians, give the opportunity to make questions to the EURCAW by this part here in whoop. Oh, it's not working. Oh, sorry. I don't know what I did. Not going backwards. Oh. Okay, sorry. In services, the competent authorities can make questions, can ask questions to the EURCAW. And what is highlighted in red, you can find the outputs I've been explaining, the reports, the webinars are online, the fact sheets, and everything. And if you want to subscribe to our newsletter, you can click on news on our webpage, and you will be receiving what we're doing mostly on every 2 or 3 months. And thank you for your attention. And if you have any question, I will be happy to answer.
Thanks a lot for this nice presentation and good overview about the reference center. I think we still have a little bit of time for questions. Are there any questions from the audience here?
Hello, stand up. I'm Sarah Fouaki from the University of Bern. Just a question out of curiosity, because I'm a lab animal vet, so I know nothing of your specific field. And I was wondering if there is a percentage of acceptability of failure in the stunning process. Of what do you find, or what is considered an acceptability criteria? Thanks.
This is subjective because legislation says that animals should be spared avoidable pain, distress, and suffering, which there's no threshold. So it depends on what the official veterinarian consider is unacceptable or not.
Okay.
To be clear, every case that is not stunned right is basically not acceptable. But we have to understand to differentiate. Is it only a single case that can happen once, or have we a systematic problem so that we have a shift towards more conscious birds, for example?
Exactly.
Sorry, that was exactly my point. So, which could be considered a parameter to say, okay, here in this slaughterhouse we have a problem, or which is, I don't know, if it happens 1 out of 2,000 animals, okay, it shouldn't happen, but it's still acceptable. If it happens in 20 animals, then this is a cutoff, and we know that there we have a problem. That was the point of my question. Thanks.
Thanks to you.
The phone. It's coming.
Thank you. Great talk, as always. My question was around the turkeys and basically you saying basically all of them having pre-stun shocks in the electrical water bath. Were the slaughterhouses that you were visiting aware of that issue, and had they done any mitigations to try and help with that? No? Okay.
I'm very sorry, Jessica. No, it's... Yeah.
That says a lot, though, doesn't it?
I think it's something that we should address in the future, for sure. Yeah.
So is it? Did they observe it? Were they aware, like when you said to them that it was happening, were they aware of it, or were they not even checking,
Some of them, they were aware, and some of them were not.
Okay.
But they don't have to fix the situation.
Okay, I think we still have 60 seconds. One question, one fast question. Then no question, then only a remark from my side. You said rabbits, 99% electrical stunning.
Yes.
That's impressive for me. I was not aware that it's really so high.
Yeah. In fact, captive bolt only for very, very, very small slaughterhouses.
But captive bolt then with a pneumatic apparatus, or?
It's manual, so...
It's a manual one, yeah.
It's something that it's for very small slaughterhouses.
And do you have any idea how many rabbits are slaughtered with this electrical stunning per hour? I don't know, I'm the honest question.
Per hour, we've seen 4,000, 5,000, the biggest ones.
Wow.
Yeah.
Oh. That's impressive.
Yeah. And when we take into account the number of rabbits slaughtered in the European Union, it's 62 million. So it's more than cattle and more than other animals. So to think about the
dimensions.
The dimensions, exactly.
So thanks a lot again for your presentation. I think we applause again. Yes, that's necessary. I think we stay in Spain. About another presenter is Angela Ramon Perez. She will talk about indicated and sensor technologies to assess pig welfare at the slaughterhouse. She is coming from the Autonomous University of Barcelona.
Right.
Thanks a lot. The floor is yours.
Thank you for the nice presentation. So, well, I'm Angela, and I'm working in a WISH project. I'm here. I'm going to explain a little about the work that I did within the project. First of all, I would like to mention or explain you what is a WISH. A WISH is the acronym of Animal Welfare Indicators at the Slaughterhouse. And the main goal of that project is to assess automatically using sensors the welfare in pigs and broilers at the slaughterhouse. But it means that the project wants to assess all the welfare that the animal had along the chain production. I mean, we will use sensors in the sensor, but we will assess what happened from farm to the slaughterhouse. So the first step is to know what animal indicators are able to assess the welfare at the slaughterhouse. So once we know the indicators, it's time to search about what is the status of the development of the technologies currently on the market. So a systematic review was conducted to identify which indicators, animal-based and resource-based indicators, are able to assess the welfare in pigs. So this systematic was done using, logging the indicators on farm and transport and slaughterhouse. But in that presentation, I will only focus on slaughterhouse. So for conduct this systematic review, the PRISMAT guidelines was followed, consulting these three databases, including these, including these terms, and this was the terms of search. The first concept was indicator, second one was assessment, third about welfare, the next one was about the place, and the last one for the species. The criteria to be select the papers were only peer-review articles written in English and published between 2000 until 2022. From initially, more than 1,500 papers were found. Only 119 papers were analyzed, and from these papers, 57 were conducted in a slaughterhouse. And from these papers, 53 animal welfare indicators were identified. We have 3 ways to categorize these indicators. The first one is where the animal problem, the animal welfare problem occurs. The second one is when we can assess these indicators, and the last one is how. For the first one, when we saw all the indicators, 16 indicators were related to the welfare problem that occurs on farm. Most of them are related to health domain. And here we can see a few examples. In the first one is a subgroup of visceral lesion that here include pneumonia, pleurisity, or white spots on the liver. And the second example is the body lesions, that ear lesions, tail length, and other problems. And the remaining 37 indicators were related to the problem that occurs on transport and the slaughterhouse. And here are both together because some of these indicators are sometimes difficult to differentiate when the problems occur. For example, in aggressions, when you see the skin lesions, you cannot really sure if that aggressions occur on transport or maybe on the slaughterhouse in the lairage area when the pigs are waiting for be slaughtered. And in that group of indicators, most of them were related to mental and health domain. And here we can see 3 groups of examples. And the first one is about the stage of consciousness, that indicators are used to assess if the pigs are unconscious when them were stunned. For example, here we can see indicators like vocalizations, breathing, or corneal reflex. In the second group, is related to handling stress. It means the behavior of the pigs against the staff. That indicators can be assessed before the transport. It means unload the pigs on the truck, or when they arrive to the slaughterhouse to unload, or maybe inside the slaughterhouse when the pigs are moved to go to the standing area. And the last one is related to stress, and most of them are biomarkers. So if we categorize in when, we can find 2 manners to assess. It was in the pigs, in alive pigs, it means antemortem, and the second one is in postmortem, mainly in the carcass on the viscera. If we take a look on this group of indicators, most of them in the antemortem are related to behavior, whereas in postmortem are related to assessing the lesions in the carcass or in the viscera. And the last one is how we can assess. From this review, most of them, 40 indicators, were assessed by a human observer. The second group was assessed using lab machine analysis. Most of them are related to biomarkers or muscles analysis. And the last one was about using technologies. Here we can see cameras, infrared cameras, and optical sensors. But here it's important to remark that in the group of the first, from these 40 indicators that was assessed, were assessed using a human observer, 12 of these indicators were assessed using technologies, but this use of technologies was like a support. It means, for example, they put a camera just to record the lesions or the aggressions, and after that they assess that recordings. It means the final output came from the human observer. Once we know which indicators are able to assess in the slaughterhouse, a second search was performed. That one was a desk search, just to know which technologies are able currently to assess these indicators. That search was conducted using the Google search engine. And here you can see all the tests that we use. The term of search was peak, and each concept that you can see into the brackets. And per each one search term, 5 pages were analyzed. So here mainly we found 2 different types of technologies. The first one was about computer vision, and the second one thermal camera. In the computer vision, there are 3 different technologies. The first one is a camera using after the stunning, just to see if the animal is well or not stunned, using the record on the corneal reflex. The second one is a result of a project 2, but is still a prototype. That it was a camera to assess tail lesions and tail length. And the last one is a company that is working on technologies able to work in a slaughterhouse, and they try to develop a technology, is a prototype as well, to assess the lung lesions, like to assess pneumonia and pleurisy. And the last one is a camera, a thermal camera, just added in the bleeding process, just to check if if for control is for monitor the killness, assessing the volume of the blood drain. So for conclude, there are 3 main points. The first one is that no matter where the problem occurs, more of them, these indicators are related to health domain. From these indicators, most of them are assessed in the carcass. The second point is that most of these indicators, the vast majority of these indicators, are assessed using a human observer. Occasionally, you can use technology, but it's like a support that I explained before. And last but not least is that there is a increasing potential and interest for developing new technologies to assess these indicators as a subjective manner at the slaughterhouse. So that's all. If you have questions, I will be happy to answer you. Thank you.
Thanks a lot for this nice presentation. We have possibility for some questions. Yes, 4 minutes. The first is just here in the center.
I think it's not working. I'm not hearing you well.
It's good?
Yeah.
I really liked your presentation. A couple days ago in the Netherlands, they released really, really disturbing footage of pigs going into the scalding tank that had gone through CO2 stunning, that were fully conscious. And it was just horrible to watch. And I know that this sometimes happens. It's almost hard to prevent that it never happens, but if we can do something against it, we should. We were thinking of installing artificial intelligence cameras in some of our slaughterhouses in the Netherlands, but you mentioned a camera that you could use to see the blood in the body to make sure the animal's dead. How does that camera work? How much does it cost? Is it really good?
Sorry. This is a thermal camera, just is to add it in the trade, just to see the volume of the blood draining. So meaning if you see the volume, maybe you can check if the pigs is bleeding to consider that the pig is killed before to go to the next steps of the chain.
Okay, so the camera just observes how much blood is out of the pig.
I mean, drain it. how much volume of blood is out of the pig.
Of the pig. Oh, okay.
Other questions? Just start behind there.
Hi, thank you so much for the talk. I'm Urja from the Royal Veterinary College in the UK. I was just wondering what indicators you think are worth looking at apart from health. Obviously, I noticed there's no resource-based indicators in there. I'm wondering how useful that would be in the slaughterhouse. And my other question was on whether you've seen any use of AI combined with some of these sensor technologies and how much more effective or less effective they could be compared to human observers.
Okay.
I've asked two questions.
I will start with the second question. So the advantages of use technologies is that you know that the speed of the chain in the slaughterhouse are very high. For example, in Spain, 700 pigs per hour are slaughtered. So it's a little bit difficult to assess each pig individually. So we use technologies. It's true that we need to first validate that technologies, that just ensure that these technologies are assessing well that is supposed to be created. I mean, first of all, we need to develop technologies, and these technologies need to be validated. And after that, you can trust really that if you see that the pig is blinked, it's because it's blinking. It's because really it's blinked. It's not a false negative. So the first question is a good question. I think that I should think a little bit more before to answer it to you. So let me think. Thank you.
Eventually, we can address it afterwards in the question and answer session a little
Exactly.
bit more in detail. I think it's not worth to discuss this now in 30 seconds. It would be a little bit too hard. No, but when there are no more questions, then thanks again for your nice talk.
Thank you.
Welcome to the last talk this afternoon. Afterward will be the Q&A session. Please take a seat. They want to start here in front. I have the pleasure to announce now from the Eyes on Animals in the Netherlands, Leslie Moffa and Kees Scheppens. They will talk about electrical stunning. The last time, a lot we have heard now about gases and mixtures and inert gases, and now we go to the electrical part. Thanks a lot, and the floor is yours.
Terrific.
First we need, okay, here it is. We have our presentation back on the screen. Thank you very much for the invitation. We are constantly in search of a more humane alternative to carbon dioxide. And we have found one. And we want to spend this 20 minutes on showing that improved pig handling is the way to go ahead with automatic electrical stunning systems. My name is Kees Scheppens. I'm a pig veterinarian already for more than 35 years. I'm also called the Pig Whisperer, and I'm working with Eyes on Animals on this project.
Yeah, you're called the Pig Whisperer because you're also an organic pig farmer
Yes.
and trainer. And my name's Kees, and I had no idea. My name's not Kees. My name is Leslie. And I had no idea that he was going to wear a corduroy suit today.
Yeah.
And I am too, so we're a little bit like the annoying corduroy couple. So please excuse the outfits. Kees, maybe you can start with this.
Eyes on Animals, for the people who don't know, is an animal welfare NGO working primarily on transport of animals and on slaughter of animals, not only in Europe, but also in Africa and also in Turkey.
But first, let's get back to why we're here today. It all started when we were reading the definition of stunning in the EC legislation. Stunning is any intentionally induced process which causes a loss of consciousness and sensibility without pain. That's the whole point of stunning. If not, we would go back to non-stun slaughter, which we don't want. Or it can result in instantaneous death. But when I was traveling through Hungary many years ago, there was a slaughterhouse that asked us for help in improving the electric stunning. But when I got there, they had switched to CO2. And that was largely because of a lot of pressure from supermarket chains, even welfare labels. And although CO2 has welfare advantages, that you can move the pigs in a group, it also has disadvantages. And what I realized is companies that were using CO2 were boasting quite loudly about the advantages, which are good, but no one was really showing what happens to pigs in CO2. So even the videos that they were putting on their website, they showed everything but the pigs in CO2.
Are you sure you want to show that?
Yeah, so in 2011, I got permission to install cameras in numerous slaughterhouses with CO2, but also large throughput electric stunning slaughterhouses. And I got a scientist to narrate the film to simply show, in a very honest, objective way, the positive side and the negative side of both systems. Because I thought if we're going to discuss it, we all have to know everything about each system. Nothing should be hidden. So this was a film we made, and since many NGOs have been exposing pigs in CO2 slaughterhouses in their countries, as recently as Australia, they published this 2 years ago. I need to warn you, it's disturbing scenes, but I think many of you know what this looks like, and I think we should see it out of respect for the animals so we can make a good decision about how we should go further in the future.
And feel it.
Okay, so I think that was enough to know that it probably doesn't suit the definition of stunning that is instantaneous or it's painless. It obviously shows a lot of panic and stress and breathlessness. To be honest, the EU already warned us in a subtle way that CO2 would probably have to be phased out and we would have to rediscuss it. But at the time, no one thought that there was an economical, viable alternative to CO2, so it was just tolerated in the hope that we'll find a future solution.
And then about 10 years later, EFSA also made a report on the use of carbon dioxide, and it said it's impossible to prevent pain, fear, and respiratory distress for pigs when you use carbon dioxide. So again, they spent 2 million euros to fund research to move away from this cruel practice to ways which are more animal-friendly, more humanely slaughtering pigs.
But the research was mainly into looking at other gases, which we think is very good. That should go on. But parallelly, why aren't we investing in improving electric stunning? There's so much potential that we haven't reached yet. Because don't we already now have a humane method of stunning pigs? Look at a small-scale pig slaughterhouse in Holland here. It's a standard situation. You have them in your countries too.
My pigs are butchered there, by the way.
Yeah.
But yeah, this is what we would say a bit small scale, manual work. Who would do 700 pigs an hour, as you said, in Spain is happening right now. So every country, every entrepreneur wants to become big, bigger, biggest, and they move away from slaughterhouses to slaughter plants. And what then can be done? Yeah, if you use electrical stunning in a slaughter plant, it will create a huge problem. Claudio already said, where's Claudio? Claudio, you already said you have to move them into a single file.
His name's Antonio.
No.
Oh, sorry. I thought it was Antonio who just told me that.
No, I know
it's
Claudio.
So. Oh, sorry.
What?
No, I just started the next video, sorry.
Okay. So this means that you have to automate the electrical stunning. Also, workers using the tongue all the time, they get tired. So you need an automatic electric stunning system, as is shown in this video. Yes? But you need to move them into a
I'll let you talk because if not, they can't hear. The video is so loud.
Okay, but you need to move them in a single file. And the slaughter plant we have been working on was actually punished for using too much of electric prodding to bring them into that single file. And the video will show what that means. Right, so a pig was maybe not prodded once, maybe not twice, but maybe 3 times. So our Dutch SPCA took away their certificate. This is not welfare friendly. This is not a humane way to move pigs in an electric stunning system. So we were faced with a challenge. We were contacted by the slaughter plant and they asked us how to make a big slaughter plant with automatic electric stunning better in order to make automatic electric stunning a humane alternative to carbon dioxide. And we did.
And that's where Eyes on Animals finally had this chance. We were begging for it for years. We finally had a pig slaughterhouse that wanted to stick to electric stunning, wanted to slaughter over 600 pigs an hour, and was willing to invest in a totally out-of-the-box design to see if they could come up with something more humane than what they already had without switching to CO2. So I got a team together. By the way, I'm the director of Eyes on Animals, but I needed specialists to help us. So there are a few butchers that help us in Ghana and Turkey with training on the use of captive bullet pistols. They're the 3 guys on the right. Madeleine is my colleague at Eyes on Animals. Case is a pig veterinarian, and I'm an ethologist. And with all of our skills, we decided to redesign the slaughterhouse, and we would be heard. So it was really quite exciting. And we had Temple Grannon also as our advisor online. And she was very excited too, because maybe you know this temple did already make lots of designs to improve electric stunning, but they never got put into practice because it was at a time when most were switching to CO2. So we started drawing, just pen on paper, cutting out, using whiteout. It was actually quite fun to be so creative and to have the license to be so creative.
Sometimes there was a glass of beer.
And we often had Temple's help online where she'd look at the designs and give us input, make sure we measured everything fine.
And then using those drawings you just saw in the previous slides, the construction started end of 2021. The Larridge facility of this slaughter plant was completely renewed. Their stunning systems were bought anew, and we could more or less say what we liked to make it a system from Larridge to the stunning, to the automatic stunner that would function. So we were giving free hand. And we found out that there are crucial factors to make automatic electric stunning systems humane. And we will talk about those crucial factors in the coming minutes.
What I have to say, though, is the design was quite good, and we built everything so it was temporary, like the sharpness of the raceway or how tall the walls were going to be of the raceway and the crowd pen. Those were flexible, so we could tweak it up with time. And we worked ourselves in those slaughterhouses. So we had to move the pigs through the whole system for several days, twice. And only by doing it yourself do you realize how like the lighting, shadows, view of people up ahead, how that can make your job so difficult and scare pigs. So we really saw things through the eyes of pigs, and we learned by doing.
We totally respect most of them are Eastern European workers who have to do this from 6 in the morning till 4 in the afternoon. I really have to say I've done a lot of difficult jobs in my life, but this was one in the top 3.
And that's why the first thing we wanted to do is get rid of the time pressure. Time pressure is a killer to animal welfare, and it's a killer to the welfare of people. And when people don't feel good in their job, they lose their patience, and that's often at the cost of animal welfare. You know, they get frustrated and stressed. So we needed to get rid of time pressure. Now, this pig slaughterhouse wanted to kill 660 pigs an hour, so it's quite a big slaughterhouse. And we thought, if you can make a small slaughterhouse humane, why can't we then build 4 small slaughterhouses in one big one? So instead of going from their system, which was 660 pigs going through one crowd pen and then going through one single file entrance to one stunner, automatic electric stunner, why don't we build 4? So we have 2 crowd pens now. There's just 330 pigs an hour going in them. And then only 165 pigs have to go through each single file. Now the employees had over 22 seconds to move each pig through the system, whereas before, just under 6 seconds.
And that is a huge difference. Be silent for 6 seconds or be silent for 22 seconds. Most people start talking after 4 seconds. So we needed, that was the second crucial factor, we needed small groups. If Temple Grandin made one thing clear to us, then it is small groups. And in the next video, you will see a big group. Large groups just don't work.
And yet the tendency in almost all slaughterhouses you'll go to is they want to move large groups because they think it's less work going back and forth.
Yeah, you get tired from walking all the time, getting 6 pigs out of the larynx.
But it slows your job. Okay, I'll watch the video. Sorry.
So there might be 20 here. Paddling around. Those are also called vocalizations. Whether they mean something positive or negative, I keep that question with you.
I just want to add quickly, Case, that what's difficult too is the guy is behind 20 pigs. He can't even reach the first pig to encourage her or him to get into the single file.
Impossible.
He's got no steering ability.
Well, here we go, with 6 in a group. The video tells the story.
The double entrance to the single files.
The guy, Hungarian guy, totally relaxed, and he now likes his job.
Okay, the other thing is for anyone who's worked with pigs, you know, they're like women, they always want a choice, they don't want to be forced into doing something specific, so you need to give them a choice. Before, you were forcing them through one entrance, one single file, and if the one pig didn't want to go in, blocking all the others. But what we did now is we installed 2 single files beside each other at the end of one crowd pen, and at the other crowd pen, another 2 single files. So the pigs had the choice to go in left or right. If an employee was here, he could avoid the employee and go into the left, for example. The other advantage of this system is that they could move into the single file simultaneously. So it still gave the pigs a little bit of that feeling that they're moving in a group, especially because the divider between the single files at the beginning we kept with large open gaps so they could see each other.
They stay social animals. So then the 4th critical factor was good timing of the bunches. Like, give this next one, the door needs to be open. Yeah, like we humans, we don't like a closed door. Pigs also don't like that. They might turn around, they might feel uncomfortable, they might feel locked in. So keep the flow and keep the doors open. And for that, in a large, not in a slaughterhouse, but in a slaughter plant, you need traffic lights. So if the traffic light is green, all the workers know pigs can come. If the traffic light is yellow or red, then you have to say, hey, I have to stop, I have to quiet down a bit, don't need to move the pigs forward, I just can wait a bit.
That we really learned by doing it ourselves, because I was responsible for bringing the pigs out of the lairage, and Case was in the crowd pen, and I couldn't even physically see him. So sometimes I thought, I'm being too slow, they think I'm ridiculous, but actually it was blocked up ahead. But with the signal, I knew, Case put the signal on green, I can come with a group of 6.
Yeah, that's why she's wearing green.
Oh yeah, okay, that's my turn. The other thing that we had to do, and this we did at the very end, because of all the plants I've been into, everyone uses those rattles. Yeah, that's just, at least in Holland and Germany. There was so much noise, but I never thought we could get rid of the noise. I'm so trained to think that you need to holler, whistle, rattle to get animals moving away from you.
Shout.
But it was so annoying. It was hard to work. It gave you a headache. It was bad for the employees, and it was horrible for the pigs. Listen to the sound that we had to work in at the beginning. Also large groups here. Yeah, so
useless.
So you know what we did? We said to the boss, okay, let's just test this out. We're the boss now. Let's get rid of all the rattlers, all the paddles. No one's allowed to scream or whistle, and let's just see. And they all thought, oh no, it's going to be too slow. The pigs won't be scared. But we wanted to try it out, and believe it or not, it worked so much better. It also made the employees calmer with the animals. So now small bunches and no noise. Just these flags are allowed in the plastic moving boards. Group of 7. Notice how they're not rushed as much anymore because they've got more time. They have 4 stunners.
And still doing 660 pigs an hour.
And maybe in the earlier video you saw that in the single file they were also using the paddles to hit the pigs. We said get rid of that. It creates noise. They use it often on the metal sides. Now they're only allowed these things that look like a toilet brush, but it works. I'll just show you the video. Okay, but to make sure that your noise level is remaining low, because sometimes people become desensitized or habituated, it is good to install a decibel meter recorder and a large screen. As a reference point, people know, oh, we're still doing well, or we're getting loud again, we're starting to get it back into our bad habits.
So then there is the last but not least, no, it's not the last, it's the second last. And that is reduce the visual confusion and the fear. And I think, who was it? I think the Catalan woman, was it? You talked about you need to improve all along the pork chain. And to do that, I actually convinced Leslie to function as a sort of guinea pig. And here she is in this single file, checking what you see as a human being. And seeing as a human being can be compared to seeing what a pig sees. So, next slide, please. What does the pig see? The pig sees another pig, you can see it here, but also sees a human being. And in the large pork production systems, nowadays, a human being on a farm is very often associated with an injection, or most times not with a pleasant encounter between pig and person. So we call him the blue smurf, the blue angry smurf. And the blue angry smurf creates anxiety, creates fear. So what you do, easily try to remove the blue smurf from the picture by putting up, again, those boards.
Yeah.
And in the end, they become higher and higher. Here, another blue smurf. A blue smurf even with a blue stick in his hand. And I think this guy is not working at the slaughter plant anymore because he was more or less unconsciously hitting every pig. Yeah. He just stood there, juk, 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 and he had pigs 660 an hour.
And at the beginning, we built a shield, just like a door for him to hide behind, but then we realized we don't even need this guy anymore. There should just be one guy moving behind the group of 6 pigs and no one in front of the group of 6 pigs. You don't need a guy to direct the pigs into the single file. They will go on their own if you give them time.
Because they have 2 single files.
So what we did is, like I said at the beginning, we made everything temporary before we set it in cement permanently. Yeah. We hung up curtains, plastic curtains all along the side, and we made it totally sealed. So the only thing the pigs could see in the crowd pen was up ahead their escape route. 2 entrances to a single file that was well lit up. No distractions, no scary things on the side. And so they went for it. They saw the escape route. It worked so well that now, it's hard to see here, but now it's permanent. So all the walls are up. These are the 2 single files, and this whole side is sealed. The pigs cannot see anyone, and the crowd pen man brings the 6 pigs in, and then he exits this door, and then he goes back in. He does like a circle all day long, but that means he also never walks back in front of the pigs, of the upcoming group. The other thing we did is we spoke with the company that makes these single files and automatic electric stunners. And we said, why do you have to make anti-reverse doors that are so solid? They look like an obstruction, as if the pig can't walk through the single file. And they said, well, we still need to keep them because sometimes pigs walk backwards, so you need those doors. But we asked them if they could punch holes in them. So they've done this now, and that works better. It's less of an obstruction because the pigs see light coming through, and pigs will always follow the light. And another thing, for those that are familiar with the automatic electric stunners, at one point the pigs have to go on a conveyor belt underneath their breast, which carries them automatically to the electrodes. But here, you can see it looks a bit like a cliff. You know, like the floor is going to end and they're going to fall down. This is the conveyor belt. It's very, very big. Stands up 25 centimeters. The other thing is the belt was white. The sides was gray, so you had a lot of color contrast. So we asked them to solve those issues by putting in a false floor. False floor is just you keep the templates going all the way to the end. So the pig sees the floor, but it does slope down. So at one point, the legs are no longer on the floor. They're carried by the breast conveyor. Everything is now gray, and the belt itself has been lowered by 10 centimeters. Not much, but it's a little bit less in their face. Now, with electric stunning, the problem before was that all pigs were prodded many times, even at the crowd pen. Now we got rid of all electric prodding. The only place we allow it is when the pigs go on the breast belt, because that's still a tricky place to convince them. But it's only allowed if the brush is not, doesn't do its job. You can't right away go to the electric prod. And we no longer have a high voltage electric prod. It's only 48 volts. We've used it on ourselves. It's like a stimulation of the skin. Here's how it's being used. So first the brush, as you see. And now they have to grab it from up high once the electric stimulation, and then back to the brush. This is where the pigs... So it's gone down from 3 times per pig to 1 time every 4 pigs. So from 300% to 25%, and we think this is quite good.
Yep.
Pardon.
So last but not least, and I will make it short,
because we're almost
Claudio or Antonio. The training of personnel is key, as we already said before, and we are also using AI to support the supervision of group size and electric stimulation. Yep. Here is one of the trainings we are performing. Next week, Wednesday and Thursday, we will again be at this slaughter plant to train the new people coming to the slaughter plant. And we are talking meat quality here as well. There was a tremendous improvement of pH value. So I would say there is no reason for a slaughter plant across Europe to not install automatic electric stunning, because the meat quality is totally comparable to whatever CO2 system there is.
Now, we presented a lot of information, but there's actually more out there. But you can find it on our website underneath Industry Tips, Slaughterhouses Pigs. We're just updating it with this project, so wait another 2 weeks. It's in English. And for any of you that work at slaughterhouses or know slaughterhouses with welfare problems that may be interested in modernizing their plant, improving it, this team that I formed, we've now created a consultancy company for other slaughterhouses if they want it, because obviously a charity like EONA cannot do this nonstop. And it's called Slaughter with Care. You can take a look at that website too if you want. Thank you so much, and enjoy the rest of the conference, and thank you for inviting us.
Thank you very much for this very interesting talk. Are there immediate now questions concerning this talk or clarification that is needed before we start the question and answer session? No questions anymore. Everything is clear. Yeah, there behind.
Hi, my name is Natalie. I work at the FSVO. And if I understood correctly, they had first one line, one electrical strander, and then they invested to have 4 at the end. Is that maybe a reason why slaughterhouses wouldn't want to change their practice? Because, I mean, 4 electrical lines, more time having to look after them, more engineering?
You need more workers, more staff to do it, but they say the CEO of the slaughterhouse said he still doesn't regret his decision because to create a pit costs a lot of money, and the CO system is a lot of money too. Plus, he says it wouldn't have been a sustainable business decision for him because he does think that CO2 will be phased out or made illegal. The price of CO2 is going up and down, and he said what he likes about this system is if one of the automatic electric stunners breaks, the MITAs, he still has 3 others, so he doesn't have to stop slaughter. So he says for him it balances out perfect. But of course for a slaughterhouse that already exists with 1 stunner, they're going to say, oh, this is too much, this is expensive. But then you can work on them with the welfare labels. Welfare labels often have strong criteria, so you can use that.
And what we heard from Australia is that in Australia, the availability of carbon dioxide sometimes is just not there. And then there you are with your pigs to be slaughtered. You can't go ahead.
Okay. Thank you. Thanks a lot. Then I would propose that the speakers come again here in front, and then we can start our Q&A session. I hope you have still a lot of interesting questions, but let's see what happens. Wonderful.
Let me have it.
Feel free.
Ja. I'll just put it here.
Ja, genau. Das reicht.
Oh.
Okay, now we've heard the whole spectrum, more or less, from carbon dioxide, inert gases, indicators, and now this improved electrical stunning, which really was impressive. Are there from the audience questions? You can freely discuss whatever you want. Online also now? A couple? Okay, then we start. Okay, you? Okay.
I have no question. I just wanted to really congratulate you because I think that is real work. I really appreciate it, also in the name of all pigs of the world.
Thank you.
One for Leslie and Keys. So did you have to use more workers using 4 stunners?
Yes. Yeah, you do need more workers. I think 7 in total, more.
You choose the microphone.
Yes, I believe it was 7 more workers that you need to do this. Yeah. But maybe you get it back in meat quality. You get it back because you get some, how do you say it? Médaillon. I can't believe I'm speaking French better than English today. You get like a recognition from animal welfare organizations, like the Dierenbescherming in Holland. You can get your Better Life quality label back, for example.
Yes. Can I? And sorry, there are also concepts which already demand that the pigs are not stunned using carbon dioxide, but using electrical stunning. So, and that will progress. That is what you promised to all the pigs in the world in saying this, but we think we are on the right track.
And another online question was, would you recommend cameras in the sealed-off driveway to monitor any mishappenings, e.g. injured animals?
So for us?
Mm-hmm.
Yeah, definitely we recommend cameras in the CO2. And I heard, but maybe the people from Spain can tell us, I heard through the grapevine that Spain is going to make camera supervision mandatory in the CO2. Is that true? Have you heard of that? Okay, in any case, in all other slaughterhouses, you have it at all the key points where welfare can be compromised. I don't know why there were never cameras in the CO2 up until now.
Yeah.
So it would be great to show that.
And as long as you do find teeth of pigs in a gondola in a CO2 system, then I think we don't have to talk much about other welfare indicators.
Just to reply to your question, I mean, there is a legislation, a new legislation. I think there is some time, 1 year for implementation, but it's to put cameras, and also in the gondola and also in the chambers for poultry, because I think there is also an issue because there is two-phase stunning in poultry, and it said that it should be up to 40 until the animal lost consciousness, and then can be increased. But nowadays you don't know if it increased before the loss of posture or not. In some animals, maybe now that they are more resistant, not. But yes, this is the idea. I have also a question. Just if I can take the microphone.
Have the microphone.
Sorry. No, I think you have done a very great job, and I think it's very good. But what I was a little bit stuck was with the small slaughterhouse. I mean, when there was the electrical stunning, that it took, I mean, it stayed there with the tongs in the head for a long time there, and then they put and changed the position, and and I wonder why so long time. I mean, if it's need or if there is, because normally it doesn't have any correlation with the duration of unconsciousness. I mean, once the epileptic session is there, they don't need to be so much time.
Yeah, I agree with you. I remember I took a course with Andy from Bristol, and he said if it's done correctly, it's instantaneous. And I also asked the Dutch, why do you do that? And they say, it's because you're there filming, and we feel like if we do it longer, then the animal's definitely stunned. So it's a visual thing.
Yep.
And I told them, you don't have to do it for us, we understand. But I think they feel safer doing it longer. But I guess it doesn't do anything bad to welfare. Maybe meat quality, but...
No, it's just maybe a more worker safety, because it's not the same if they stay for 2, 3, or 4 seconds, that not for 15 seconds for each without any need. Yeah. Maybe this.
We were standing there, and he just wanted to do it very good.
No, we have faced also this situation that they want to be sure that the animal is unconscious, and they stop for a long time.
This was quite a few years ago for me now, but holding the tongs on, from an engineering point of view, I'm an engineer, it heats up. Yeah. That creates a sort of grease or whatever, a film on the electrodes. So the next time you use them, you get reduced current because they've got electrodes. So don't hold them on too long, is the answer.
Okay. We'll definitely tell them that, though.
Okay.
Hello, Charlie Mason from the Humane Slaughter Association. Again, it's another question for Leslie and Case. You said that the only handling aid allowed now is the brush, apart from the occasional use of the 48-volt thing, which I've experienced. When I was over with Roy in the abattoir that he used to work at, where he prodded me with the 48-volt thing, they were using an inflated bin bag, and they walked behind the pigs doing that. Oh, yeah. They didn't hit the pigs with it, yeah. And the pigs moved. Yes. Why are you not using that? Because I thought that was one of the best things I've ever seen.
You mean like an inflatable...
They got a bin...
Inflatable plastic bag.
Got a bin bag, put the airline in it, and they walked behind the pigs doing that, and it went bum, 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 bum. It didn't touch the pigs.
Yeah.
But the pigs...
They still use that, but only at the unloading.
Ah.
Because they're moving larger groups off the truck, but we thought it still creates some noise, and the plastic flag on the little rod did the trick as well and wasn't creating any noise, because when you do this, you still get that sound. But you're right, that's also an ethical tool. I mean, it doesn't hurt the pig.
Hi there, Josh from Vet Record. I've got two questions really. One is, what were the challenges in persuading the slaughterhouses that Eyes on Animals should be in charge of the killing of the animals in the slaughterhouses? And also perhaps there were challenges within your organisation, Eyes on Animals, in persuading your people and your supporters, we're going to now be killing all these animals, and why you should do that. And also, can I just check, Leslie and Keys, are you saying that the CO2 gas stunning, in inverted commas, should actually not be considered stunning. In other words, it should be considered non-stun slaughter, because this is something I hear a lot whenever we write in Vet Record about non-stun slaughter, meaning the halal and kosher slaughter of animals. People from those communities will always come back and say, well, what about CO2 stunning of pigs? That's not stunning. So why are you saying, we can't kill our animals by non-stun? Yeah. So are you saying it should actually be reclassified as non-stun slaughter?
Ooh. I have that experience all the time because we work in Turkey to teach captive bolt stunning to those plants that are open to it. And they often say that. They say that they feel very accused for being cruel to animals because they're Muslim, but what about the Christians that are forcing pigs to breathe in CO2? Now, I think all cultures, all religions have good and bad practices with animals, and I agree with them. I think the way we stun pigs, it is cruel with CO2. If we have a better alternative that's immediate, then we should go towards that, or at least get the full potential out of it. But I think they have a point in that. The purpose of stunning is to render bleed out and cutting painless. But if the stunning itself is causing a panic attack and breathlessness and acidity to the mucous membranes, then I think we're defying the purpose of stunning a bit.
Yeah, I now talk as a pig farmer, and I might have a little bit more of a knowledge of how pigs are slaughtered, and I won't send my pigs to a slaughter plant where they are using carbon dioxide to stun pigs.
Can you answer about
Yeah, the first question, okay, what do donors think about a charity doing this? Yes, of course, I've lost donors because they think we're helping kill animals, but we also have donors who understand that the world is not going to change overnight, and every step to reduce suffering is wonderful and is realistic and is attracting the mass of society, not just the ones on the side. And I don't design ISO animals according to the donor's wishes. I design it based on what I see in the field and what I think can really make a difference. Was that the question? Yeah, that was it. Okay. And oh, by the way, with the slaughterhouse, we just got lucky. I'm Canadian, and what I loved about moving to Holland is that you can sit at a table with the CEO and chat and have a coffee and discuss very openly. And this was just a slaughterhouse that was really friendly and accepting towards us. We're not very radical. But what really helped was the Dierenbescherming. That's the national Dutch welfare organization that has the Better Life Quality label. They threatened to remove it if they didn't take serious action. So that was our access door.
And the general manager of this slaughter plant, in the beginning he said, those animal welfare, and then there's a word starting with a B and ending with a T. But in the end, he now really is extremely happy because he can show to everybody in the world how automatic electric stunning is a way that at least respects the integrity of every pig and not brings pain and fear and distress to every pig like carbon dioxide is doing.
Thanks, Hugh Gollidge from UFAW. It's a question for Jonas, actually, going back a bit earlier in the afternoon. You've demonstrated that the engineering challenge of switching to inert gas is actually not very significant. I suspect it might be a very appealing switch for the industry to keep using gas stunning, but to find a more humane version if we have to stop using CO2. So what do the industry tell you are the barriers to adopting inert gas stunning?
I think, main challenges will be costs and just argon costs, but I'm not 100% sure with usage, and just that the exposure time is a little longer. So that, of course, affects how many pigs you can stun per hour. So there are some challenges, but my feeling is that once some slaughterhouses start, there might be the incentive for others to follow.
Because I think the argument at one stage was that argon was going to be dramatically more expensive than CO2, but actually that may well not be the case now, and the supply may also be reliable, which might break down some of those barriers. So let's hope.
When we just stay with these gases, what are now your realistic plans? What's going on? What are the next further steps? And could you already give some recommendation?
I I can't.
I'm
not sure if I can make a recommendation here. So for us, our next step is that we hope to verify our results in a high throughput paternoster system, where we would use argon without any CO2.
Okay.
Argon mainly because it's easier to handle than the nitrogen, and not necessarily because that's worse, and because we didn't see a benefit of adding CO2.
But then really on an industry scale, high throughput.
Yes, I mean, this was already but it was small industry, but it was a commercial dip lift system that is being used every day. And then for our experiment days, we ran it with argon.
Okay. Okay, if I look in the audience, we still have other speakers here. Yeah.
Hello, it's Charlie again. Question for Angela. In one of your slides towards the end said that the majority of AWIs were down to health.
Mm-hmm.
Presumably that was based on the post-mortem results. So does that mean that majority of those were not associated with transport and slaughter, but more to do with husbandry?
I think I don't get you the last part of the question. Can you repeat in another words, please?
Yeah, you said that the majority of the animal welfare indicators were associated with health.
Mm.
So, and presumably they were exposed at post-mortem.
Mm.
So there'd be lung damage, things like that. So does that mean that they were more to be associated with husbandry on the farm than recent injuries in transport and slaughter?
Depends on the indicator, because if you are checking the ear lesions or tail lesions, I suppose that these lesions occur on the mainly on the farm, because that the type of lesions that you see in the tail or ears is like chronic. And for example, if you are seeing the scratches or skin lesions, you cannot differentiate if that lesions occur during the transport of the latest area. It's true that you can see the evolution of the lesion, but maybe it's going to be so specific, because depends if you has a fresh blood. Maybe you can see, you can tell like, okay, that maybe occurs on the latest area, and if you have the most chronic, I don't know how to say in English, no, not abscess, like
lesions.
Like healed, right? That's what you want to say? Costra. In the process of healing?
Sorry. So that one, maybe you can say, okay, maybe if the travel occurred several hours ago, maybe you can differentiate. So that is depends on the indicators that you are assessing. So I'm not sure if I repeat your. Yeah. Okay. Thank you.
Okay. Other questions? People seem tired, but we still have time. Okay.
Yeah, okay. Now, I don't work with pigs, but welfare is also about the emotional state of the animals. I was very surprised the pigs can see the other ones dying, and because of the cognitive abilities of pigs, I'm sure they do perceive pain, well, like kind of the stress of the process itself. So how could you maybe improve on this? Have you think about like a way of not having this contact while the animals are just kind of dying?
I think, Case, because you're a farmer, you're more skilled to answer that. But as a welfarist who's seen a lot of slaughter, I'm not so worried that pigs can see other pigs being killed as long as the atmosphere is calm.
Yes.
I don't think that they really know, hey, my friend is getting killed. I don't even know if they know what that all means. I've seen pigs actually enjoy licking blood. I think they sense much more the tension in the room. so that's where we try to focus on, reduce the tension and the speed, and the rest, yeah, it's not such a priority, at least for me, for
No.
what I think is necessary.
I would agree. An experiment at younger age I wanted to do is to bring pigs to a slaughter plant and then not butcher them, but bring them back to the farm and then bring them again to the slaughter plant. I think then there might be a recollection of what they heard, what they smelled. I don't think we know exactly what they can really smell in a slaughter plant. And then I think there might be some start of seeing what might happen to them. But when you bring them there the first time, we have seen thousands and thousands of pigs. I don't see that there is any, let's say, preconception of what might happen to them. But I'm only a vet, so. And if two agree, then one is not a vet.
But I can really confirm this. I have the same experience with cattle also. You can shoot away one beside and the other is still eating and not moving, not interesting.
Yep.
It is the environment where this happens, as you said. Really.
Thank you. So this is a question for Case and Leslie. A lot of the recent animal welfare research has been looking more into soundscapes, so the effect of music or of different types of sound environments. Do you think something like that would potentially be beneficial within a slaughterhouse, so potentially masking other sounds in the environment?
I have my doubts about that because still the noise level is pretty high in a slaughter plant.
Mm-hmm.
There's always metal on metal. You try to avoid that as much as possible. But for example, the moving boards, sometimes they use them to just hit the board on the floor. So I don't think that there is a lot of influence to be expected from Mozart or Beethoven or even Rammstein, I think. Maybe Rammstein would do it.
Yeah, I don't know if I fully agree because there is a slaughterhouse in Germany that has even made their own cassettes and they call it Pig Pop. And they think, they don't have proof, but they think that it makes the workers happier. And because it's melodious pop, that it kind of blocks out the peaks of other noises that may frighten pigs. And they did do studies on dairy cows that they drop their milk often more quickly if there's classical music. And I have been in a really bad slaughterhouse, electric stunning manual in California, where they were blasting salsa. And I think just that blasting music scared the crap out of these animals. They were really scared. So I do think you can play around with sound. Nice sound is obviously, I think, going to create less stress than abrupt noise. So you could play around with sound to take out the peaks, I think. It's interesting. But what would also be interesting is smell, because pigs have such an incredible smell. For example, could you block the smell of CO2 so it's not so aversive, so it smells like apple pie or something? I don't know.
Thanks for all your questions. There's still something that needs to be discussed. If I look here, the crowd. Sure.
Yeah, we have an online question for Jonas. How easy is it to convert existing CO2 controlled atmosphere stunning to inert gas mix systems? And what are the costs associated? And how does the cost of the gas mixture compare to just CO2?
Well, I can't tell you how expensive it would be to install that. Installation is fairly easy, I'd say. As said, for both the installation in the dip lift system and the paternoster, it was roughly done on one day, installed, and after that it was functional.
Other questions online? While you are thinking about more questions.
He has a question.
Okay. Sure.
I'd like to ask you if you also looked at the stunning effectiveness, because that's like for me always has been like the other issue with electric stunning, which also showed up in field studies and was my personal experiences, that there are sometimes bad days, and when there's a bad day electric stunning, then all the animals end up gasping at the end, and if you follow them closely, regain consciousness, briefly at least.
Yeah, absolutely. At the beginning, we felt like there was a bit of an issue, so we asked the company to come back and clean everything and check everything, and then it was going much better. So I think maintenance is really important, keeping it clean, like what you said with the grease or the buildup of burnt skin or whatever you want to call it. But what we were really concerned about at the beginning was we did see some pigs regaining consciousness, but it was because the sticking was really bad. So they were stunned properly, but you only have like 15 seconds, although they were heart stunned too. So that should have been enough. But still, the regaining of consciousness was happening because the sticking, the guys that were sticking had not been properly trained. And so not enough blood was coming out quickly. But they've now received extra training in sticking, and it's a lot better now. The carcasses are hanging quite limp. And at the beginning, you're always unsure because some carcasses are moving a lot, and you don't know if it's a sign of regaining consciousness or not. That's not so easy. But now that the sticking is better and that the electric prods have been properly maintained and the parameters changed a bit, it looks really good now.
Yeah,
but it is a big problem in most slaughterhouses. That's true.
So in the beginning, yes, there were some issues, but now there is extra. Also, there is extra control from the state veterinarians. So we are using artificial intelligence cameras to check that every pig really is dead before the scalding tank. Yeah, so the videos which you see on YouTube last week, they were actually from Belgium. They were not from Holland, but they are also from a carbon dioxide plant.
Antonio? Yeah. Now the real Antonio.
I have two questions. One is for you, follow up what you say, about the camera. Yeah. How the camera says that the animal is dead and not unconscious, but still alive. This is the first question. I have another for Jonas, but I don't know if there will have time.
But what's the question then?
Because before going to this, as far as understood well, that maybe I'm wrong, to ensure that the animal is dead before the scalding is using cameras.
Yes, yes.
And the question is, what is assessing these cameras to ensure that the animal is dead and not unconscious but still alive?
Yes, you can do it.
You said it, but I have to defend it. Well, at the moment, the artificial intelligence is picking up like uprighting movements of the head or a carcass that is performing really abnormal behavior compared to the standard, so one that's moving a lot more than the others. But we have a person there, and that person's responsible to re-stun right before the scalding tank. And the camera is just like a backup to get figures at the end of the day of how many carcasses are acting abnormally compared to the others. Because at the beginning, there were too many carcasses, with our feeling, that were moving, that were not as limp as they normally are 2 minutes after bleeding out. So, but I was more interested in the camera that the Spanish girl was talking about that was looking at how much blood was coming out. And I know Turnus in Germany actually measures the amount of blood coming out per animal before they go in the scalding tank. That's probably expensive, but that would probably be the best. But I have a question for you, Jonas. I'm going to get you back. Now that there is no such thing as a perfect slaughter. But if we have to have high throughput and not small slaughterhouses, which I think can be humane, if we have to have larger slaughterhouses, based on what you've seen in your studies with nitrogen and argon mixed, and the videos that we showed you, do you feel like one has a little bit more potential than the other, or would you say they both still have their disadvantages and advantages? Or if you were a pig, where would you want to be?
Well, it's very difficult to answer, I think. I think from yours, I would need to see more data, actually, to actually see how reliable that is, to make that judgment. Right now, I wouldn't say that inert gases is the perfect system. And from what I've seen from electric stunners after ejection, the issues there where often the electric current for the heart is not enough to actually induce fibrillation. I would also not say that electric is the best option, even if the issues before are solved, which does look pretty good. So I can't say that between those two I wouldn't be able to tell. Right now, for me, both likely are better than than CO2. Yes.
I wish that these companies that make these systems, like the company that makes the automatic electric stunner, the companies that make the CO2 systems, I wish they were to do these studies more often, because it's bizarre. We've had these systems in for years, and we're still unsure if they're working so well.
Okay, last chance for one
yes,
question.
I had a second
Yes.
question for Jonas, because you said that you have done the studies in the pilot and now you are going to scale in commercial conditions. And I would like to ask you if you expect any change. I mean, because there will be the animals moving to the Putina, to the deep leaf. I mean, there will be exposed more people or the exciting. Are you going to consider some these external factors in your assessment? Because it may influence also them.
It can influence. I think you can have a dip lift system where the animals enter it very stressed, and you can have a paternoster system where they enter it very stressed. So I wouldn't necessarily expect a difference there. From the once they entered, of course, if they're very stressed, that can have probably have some influence. But after that, once they're in there, I don't expect too much of a difference. Actually, of course, they should get in there as calmly as possible. But I wouldn't expect lots of differences there.
Okay.
Will you take into consideration also the pre-handling factors? I mean, the transport, the fasting.
It's tricky to do it at the exact same level that we did the rest of the study. So for us, everything we did so far, we were tracking the animals individually. So from entering the stunner to the meat quality, we know which animal is which and which one showed a single gasp after ejection. And that's, of course, becomes unfeasible before that. But there are some plans in another project, part of the Pixan project, where that's supposed to be looked at, but likely not at that level of detail.
Is there something open online? Nothing? Okay, then I would say with that I conclude this session. I don't know if you want to say something. No, then let's say like this. We continue tomorrow at 9 o'clock here again. I wish you all a nice evening, but first I really want to thank all the presenters for their really outstanding talks. It was really interesting. And I want also to thank the audience for all the support they give us here. And please come back again tomorrow. The presenters, please stay seated here. You get a little gift.